It is with great pleasure that we extend a warm welcome to each and every one of you to this 5 days online international faculty development program on bridging research and innovation in biomedical sciences and engineering, organized by School of Allied Health Science, School of Biomedical Sciences, and the Institute Innovation Council, Galgotia University. We are highly privileged to have assembled a distinguished panel of speakers and experts who will share their expertise, experience, and latest advancement in the biomedical field. And this program presents a unique opportunity for all of us to learn from each other, exchange the ideas that will propel our collective pursuit of excellence in research and innovation. As a participant, your active engagement and enthusiasm will play a crucial role in making this program a resounding success. So we encourage you to actively participate in all the session, ask your query, and share your insight and experience with us. Well, it is my honor and privilege to introduce our esteemed guest speaker, Dr. Manisha ma'am. She has joined with us. Dr. Manisha Singh ma'am is currently working as a research fellow, discipline of pharmacy, graduate School of Health, Faculty of Health, Australian Research Consortium in Complementary and Integrative Medicine, University of Technology, Sydney, Australia. She is a highly motivated and responsible researcher and academician who enjoy the work and strive for the excellence. Her research interests are in nanomedicine, pharmaceutics, and targeted drug delivery. Currently seeking suitable opportunity, and she has also worked as an assistant professor, Center of Emerging Disease, Department of Biotechnology, JP Institute of Information Technology, Noida. She has also worked as a research scientist in the Molecular Dementia and Memory Research Lab, Flinders University, Australia. She is also a lecturer in the Biomedical Engineering Division, Department of Biotechnology in JP Institute of Information Technology, Noida. And she has contributed in the clinical setup in handling the neurohabilitation patients in OPD and IPD facility in various organizations such as IMR, Stephen Hospital, Apollo Hospital, and PIP RMS Hospital. She has organized various services camps in remote area in Delhi and NCR for underprivileged community group under an institutional community awareness program for the epilepsy as well as several other disorders. She got awarded and conferred various awards and recognition, including Conference Fellowship for the Alzheimer's Association International Conference. She also awarded with the Global Talent Subclass 858 under the Global Talent Independent Program. She received Excellence in Neurosciences and Nanotechnology by Healthcare Leadership Award. Also got a Young Research Scientist in the Field of Nanotechnology Award, awarded with the travel grant by IEEE Photonics Society, A+ grade certificate course on basic neurodevelopment techniques and best rehabilitation intern. She is also awarded with Best Student Award for the academic achievement in graduation course, Best Cadet Award, High Commendation Award, and many more. So that the award she has received are countless. So she has published more than 50 plus research and review papers in reputed journal and presented her research in various international and national conferences. She also authored books and book chapter with reputed publishers, and she is the member of numerous scientific societies and delivered a number of invited talks across the world. So we are very fortunate that ma'am has accepted our invitation to deliver the keynote talk. So we are welcome you, ma'am. Over to you.
Thank you, Dr. Gaurav. Thank you for the introduction. And should I share my screen?
Yes, ma'am, you can.
I hope it is visible to everyone.
Yes, ma'am, it is. But it
Okay,
is not
very good.
in the presentation mode. Uh,
Okay, I
yes.
had to switch it to presentation mode. Okay, it is visible now.
Yes, ma'am, visible. It is visible,
Yeah,
but
so
not in the full screen mode. It is.
still
It will
not
be
there.
better. No, no.
Okay, and
In the below panel, there is a zoom in and zoom out.
yeah,
Yes, ma'am. You can reshare, ma'am.
yeah, I'll reshare it. Um, is it visible now?
Yes, ma'am, it is visible.
Okay, so shall I start?
Yes, ma'am. Please.
Okay. Thank you. Thank you. I would like to extend my thanks to the organizers of this FDP, and I hope I can share some insights about my field of research and my field of working with you. And so today I'm going to talk about the translational research in CNS drug delivery, why it is important to look for specifically for the translational research in CNS disorders, because the CNS pharmaceuticals with nano formulation and targeted drug delivery strategies are the upfront things which are coming along with the translational research in this uh, area, basically. So, if we see the background of it, background story, if I just can sum up in a short way, then we all know that the therapeutic areas, disease of the CNS provide the biggest challenge to translational research, especially in this era of increased productivity and novel targets. So the risk reduction by translational research incorporates there is a very known paradigm that has been shared in the industry that says that the learn phase of the learn and confirm paradigm, which was proposed over a decade ago in the pharmaceutical industries. So most of the pharmaceutical industries, they actually didn't want to venture out more into the CNS disorders, because most of the things at the molecular as well as at the cellular level were not basically explored in the CNS disorders. So that led to the delayed in the pharmaceutical interventions. And in current state also, we hardly have the medicines for the diseases like your meningitis or the neurodegenerative disorders like your Alzheimer's, Parkinson's, whatever we have, we just have with the symptomatic perspective. So that again leads to the necessity to develop more therapeutic things which are more meaningful and which are more efficacious for the CNS disorders. So moving ahead with this research hurdle further, let us explore the need of varied therapeutic strategies to deliver efficacious treatment, especially in CNS related disorders. Now if we see, if you see this, basically, we are aware that CNS disorders are broadly categorized under either the neurological or the mental disorders. These are the two very broader categories under which any of the neurologist or neuropsychiatrist, they will categorize the patients. So, your neurological disorders, they basically range from being the dementia, Alzheimer, Parkinson, epilepsy, headaches, various types of it, to the various neuromuscular disorders. However, your this mental disorders are the blend of the diseases like your psychotic diseases, the mood, anxiety disorders. There are many other psychiatric the mental ability related disorders. So basically, if we just go and dig much deeper into the actual existence of each disease, CNS diseases, then we further look on the global scenario that the previous years, from last 2 decades, the United Nations and World Health Organization WHO basically publishes after every alternate year the the disease burden of various CNS related disorders on the global community. And they share that the public health challenges that shows that people in all countries, irrespective of their age, sex, education, or income, are affected majorly by the economic are affected by these neurological disorders. And the economic cost of such disease, in especially if I talk about the Europe, has reached to 139 billion euros, which, by it is as per the report by 2020, which was been published by WHO. And that is an alarming thing that was being raised on the global platform. And it was also been seen that neurological disorders are the leading cause of death and disability in the world today. It is not only the death, rather it is more of the disability scenario that has been the people or the research community is more worried about. So it is, as per the reports in 2021, they were ranked as the leading cause of group of DALYS, that is disability adjusted life years, and it comprises comprising of about 10.2% of the global DALYS. So again, seeing that is this entire scenario is quite alarming. And the most prevalent neurological disorders of them all, if I leave aside the mental disorders, which is also a major contributor to the global economic burden, but if I talk about the neurological disorders specifically, here the leading cause of death and disability is more prevalent neurological disorder is your Alzheimer disease and other types of dementia which are associated with the Alzheimer type symptoms. So somewhere about 46 million cases are being registered and reported around the globe in the current scenario, and that has been further followed by the tension type headaches, then the migraines, the medication overuse things, and your epilepsy. And then there are many listed problems which are already there. So uh, if I talk about the our own country, India, how we are calculating on these risk factors. So after the publication of these research and reports on the global scale by the UN and the WHO, even the India did the comprehensive neurological disorder surveys throughout its all state, and it was the first ever comprehensive study which was been planned by the government of India in the last decade. And because of the same reason that these disorders are increasing on a very alarming rate on the global scenario. So the government of India, with its Ministry of Health and Family Welfare, joined hand with ICMR, Indian Council of Medical Research, and Bill and Melinda Gates Foundation, all three of them collaboratively did this first comprehensive study in every state of India from 1990 to 2019, and they saw the steep growth on the in the neurological conditions. What is the count of the people that is there in every state? What are the economical backgrounds they belong to? And it was a much deeper study that was done, very much needed also. And this entire study suggests the need to steer the health system to address these neurological disorders specifically in a more effective way. And this report could be useful as a reference in the future planning of approaches to contain the increasing burden of these disorders in the all parts of the country. So, what was the result and outcome of this? This particular study demonstrated that in India, since 1990 till now, till 2019, the total neurological disorders or the non-communicable neurological disorders basically doubled from 4% to 8.2%. This I'm talking about 2019. And then it is increasing on a very higher scale. So again, the other we can expect even the threefold high in the next report that would be coming up. And what was were the contributing factors to the neurological disorders in India? Here, the scenario is a bit different than the other global or the European countries, and it is due to mainly due to the high systolic pressures, blood pressures, pollutions, dietary risk, high fasting, plasma glucose, maybe the mainly the diabetics. So these all are the factors which contributed in the total count of the patients there in India. If I talk about the CNS pathology, like what we expect in CNS, I mean, you know, what all are the combination of pathological problems there in the brain? Forget about the Alzheimer, Parkinson, or epilepsy or psychotic disorders, but what exactly may go wrong inside the brain? So that is the most important thing. And if you observe the brain and the major CNS pathological incidences that causes or irritates or initiates the NDDs are basically the process of neuroinflammation. That is the first most thing with most of the CNS disorders, wherein there is an elicitation of INOS, TNF alpha, IL-1B, COX, NF kappa B factors, all the inflammatory factors which are in any way in if you get the inflammation in any part of your body, these all chemokines and cytokines they get elicited over. But these factors, why they are important with the CNS disorders are they because CNS system is in closed circuit and it cannot be accessed so easily with either of the therapeutic interventions as you may access it throughout your body. So this is one thing, and these cytokines, chemokines, all they lead to neuronal damage or maybe microglial activation also, and they disrupt the BBB along with, and this entire thing cause the neuronal dysfunction. Now this dysfunction can be of various types, like due to the aggregation or deposition of these proteins or your amyloid beta proteins, synuclein, NFTs, neurofibrillary tangles, you may get the Alzheimer. If not, then your body may respond in another way with the elicitation of the Parkinson. Then, if the physiological circuitry system or the excitocircuitry system is in the toss, then you may have the epilepsy symptoms. So, the way brain reacts with all the protein accumulation and which part it hits it, maybe it's the neurons or the microglial cells or the activation or the pathway or the transmission, no electrical impulse transmission, what has been hit, what has been compromised, elicit the type of disorder. So the other physiological changes like your lipid peroxidation, astrogliosis, DNA damage, this all is the part and parcel of your pathology that may occur inside the brain. So all in all, this all thing can happen inside the brain, and we had to target that how we can compensate, or how we can directly or in a very targeted manner can go for the treatment of that. So out of them all, in one way or the other, most of the diseases like your epilepsy or meningitis or other cancer gliomas, in fact, they have the possibility of surgical interventions. That is, through that process, like your stroke or your many other complications there in the neurological disorders can be still taken care up to certain extent, not completely, but up to certain extent through the surgical interventions, or maybe the symptomatic targeting also works with the medicines. But there is one dreaded disease, that is dementia or the Alzheimer disease, which till date, even it has been explored, discovered 5 decades ago, but still we don't have any cure for that. Neither we can we have the symptomatic treatment. I mean, in a way that we can at least cover up the 40-50% of the disease. It is just very like only 5 drugs in the market till date, and still they cannot improve on the condition, or at least stop the Alzheimer condition. It is the sixth leading cause of death in European countries, and in US, UK, and European countries. It is the fifth leading cause of death in the age 60 till the age of 65 and older, and also the leading cause of disability and poor health on a global scale. And it has been estimated somewhere around 24.3 million population globally registered and reported. God knows how many more are unregistered and unreported. India basically is having around 1.6 million affected registered patient here. So this is something, again, a very grim kind of situation that is happening. And if we see the world's biggest killers, out of them, the neurological disorders are at the fourth level. Though the cancers in brain have also elevated at a very larger scale. Uh, if I talk about the country of my workplace, there it has been seen as the disease that is causing a economical burden of $74 billion every year. And the people with the dementia is predicted to triple in Australia by 2050. Somewhere around 90 million people would be affected by this. So for them, yes, it is the disease that they are looking forward to find out a particular solution to it. What can be the solution to it? Awareness is the key to tackle. Now we are lucky to have been in that era when most of the molecular pathways and most of the cellular changes are being already discovered, and we at least 80% are aware of what exactly goes wrong with the most of the CNS disorders. So if we talk about the translational kind of work, that what is a translational approach? So basically, it's been seen that in the pharmaceutical industry, translational research refers specifically to those activities conducted to bridge the gap between the drug discovery in animals and drug development in human patients. So this is an industrial version of translational research or approach. So the need for such activities has been driven by two major changes in drug discovery, that is increased productivity, first thing, and the focus on novel drug targets not yet pharmacologically proven in man. So you have to take up a drug target that can hit the pathological site directly. So this is the very clear scenario in the industry. And since why the industry was not very keen and motivated towards investing in the CNS world or CNS disorder commodity, because they neither knew the pathological target very accurately, and though they have the novel drug targets, there are many in the traditional complementary system of the medicine, and even in the synthetic or the allopathy system also. There are many drug targets which can actually hit and solve the problem of various CNS disorders. So it's also been seen that only 3 to 5% of CNS candidates ever become the marketed therapeutics. So this also, demotivates the industries to take up the CNS things. So in the larger sense, translational research could be taken to encompass the entire breadth of medicine, right from the diagnosis part to the prognosis and the management altogether. Also, many available medicinal things are rendered ineffective in the treatment of CNS due to inability to effectively deliver and sustain them within the brain. Like uh, you may be having a nuclear weapon, but you cannot throw it on a targeted site. Then what is the use of the same? So it is the similar kind of situation that is happening in the CNS world right now. So it has been seen the clinical failure of potentially effective therapeutics is often not due to the lack of drug potency, but rather to shortcomings in the method by which the drug is delivered. So this is the major gap factor. Um, and therefore several active agents may be abandoned because sufficient drug concentration in the brain cannot be achieved by blood. So under physiological conditions, substances may cross the BBB via transcellular lipophilic diffusive route. There are many things associated with the delivery system also. So one such very important factor is your blood-brain barrier. We call it as BBB. The thing that you are seeing on the slide is the exact arrangement in the CNS system, which is a very close packed compartment, and it doesn't let anything go inside the brain without the surveillance. And BBP represents a major challenge for the drug delivery into the CNS microenvironment. And the presence of tight junctions and other specialized cells in the barrier, it retards the paracellular flux of intended therapeutic molecules and by posing high electrical resistance. So basically, it basically stops everything that even if you send any like since ages we know that curcumin is a miracle substance compound, and that can basically treat most of the neurological complications, especially if I talk about the Alzheimer. So it has been there in our in our ancient traditional medicinal system also. It has been reported in allopathy also has validated that yes, curcumin is one such very important compound. But the problem lies that we can't cure the Alzheimer with curcumin till date, because again the target, the drug, the pathological target where we want to hit it cannot be easily achieved due to this BBB, that is blood-brain barrier, or the blood CSF barrier, the another thing. However, the lipophilicity is a double-edged sword, which has been needed because the drug delivery to the brain is preferentially attained by small molecules with high lipophilicity, as they can passively transverse the BBB. However, lipophilicity is a double-edged sword, as highly lipophilic drugs they result in the poor absorption. Like it is very common about the curcumin that it cannot be dissolved in uh, any of the aqueous solutions. So you have to make some preparation, some formulation preparation for that, so that it can be solubilized and it can be delivered. So it has a low solubility and fast metabolism. So small lipophilic drugs that are less than your 500 kDa size, basically they exhibit very low therapeutic, more than 500 kDa, they exhibit low therapeutic effects due to single molecular targeting. So although most of the macromolecular drug, including therapeutic genes and any mRNAs, which are very potential to target the pathological sites in CNS disorder, they cannot be done due to their size range or high molecular weight, which cannot permeate through the BBB. So the limitation of today's modern day medicine is what what it shows is that they doesn't have holistic approach towards the treatment of disease or the disorders, and they provide more of a symptomatic treatment which has a suppressive approach for the disease. That means if I put it in this way, that the allopathic medicines has one disease, one target, one drug strategy, not the holistic approach. Secondly, certain enzymatic action may convert them into non-therapeutic intermediate compound, leading to its side effects. And also, if we see the major reason for therapeutic failures, it is due to the drugs show slower drug action due to BBP, as less content and slow permeability through it prevails. So then, depending upon other solubility, molecular size, weight, absorption in the gut, there are many other factors which gets add on. Right. So these are the first-hand things related with the problems associated with the CNS disorders. Now, if I say that how we should approach, now the question comes that okay, fine, we cannot send everything to the brain because you have a very high surveillance system over there. So how you had to approach the brain drug delivery? Now, there are the three broader categories. One is the invasive, non-invasive strategy, and advanced strategies. So, invasive strategies comes all your surgical interventions like your craniotomy, microchip technology, and even the AI simulated kind of things which are nowadays very in, and AI guided surgeries which are being done. So it is more of the medical thing that is, but then they have very high risk factor also associated with it. In the non-invasive strategies, we have plenty of examples, plenty of options. Like we can design the nano carriers as drug delivery system, or we can also opt for the different route through which we can deliver the drugs. Like the one very famous sort of delivery system is intranasal drug delivery system, which may be new to the allopathic world, but for the Indian medicinal system, this was already been reported and well listed in the Charak Samhita and Ayurveda. It has been listed as the Nasikriya treatment, and most of the CNS disorders were that time being treated through this route only. And if I give you another very simple example, that the time when the situation comes for the cannabinoids and all, um, or the drugs, when the person takes it, they sniff it, so they get high very easily when they sniff it. It is much um, it is much on a very higher scale when you sniff it rather than you just take it through oral route. So basically, the intranasal drug delivery system, or the intranasal route, is the direct window to the brain to sending anything. But then it also has certain good specification what kind of stuff can go through this route to the CNS system. And then the recent advanced strategies are, we are not yet with the commercial product of these strategies, but there is a high chances of these strategies, like your antibody mediated drug delivery systems, or your genetic modulations, or the CRISPR-Cas9 technique that has been coming recently added, can help a lot in delivering or treating the diseases from their root cause. So these are the different strategies through which the brain or the CNS can be approached for. And then we can see the light for the translational thing for CNS drugs. Um, CNS delivery, basically, if I say, then it can be done in a various manners. People are trying it through different ways, like with the endogenous biology. Endogenous biology means through the transporter system, like you are impregnating the tight junctions which are there on the periphery of the BBB or blood CSF system. So you just pick and select those transporters which can go or permeate through that, through this system, like your extracellular moieties and all. Then receptor mediated transcystosis also, because we understand that transcellular transport also goes through which the nutrients are being passed on or the blood circulation goes on. Then the other very important strategy is adeno-associated viruses. Though we do have certain questions about this virus related uh, virus mediated kind of therapeutic thing in the brain, but till date, adeno-associated viruses had shown great promise in delivering the drugs to the targeted site or the biocompounds to the targeted site. We have mechanical disruptions, but again, compromising on the BBB or blood barriers, CSF barrier system would be too risky for treating the any kind of CNS disorders. Like it can be done through the osmotic or the mannitol thing, and then the peptidomimetic system or the focused ultrasound through which the drugs are being reinforced inside the membrane. But it basically at times, if you don't go beyond the limits, then it is reversible phenomena. But then remembering that your CNS cells are too fragile to bear the higher altitude kind of disruptions. So it may at a certain time point may become the irreversible change in the brain. Then the most promising one are the bioengineered kind of biomaterials like your liposomes, nanoparticles, extracellular vesicles. These are the vehicle systems, or these are the carrier systems which basically encapsulate major therapeutic biomolecules or maybe the synthetic compounds inside them, and uh, they can impregnate the BBB and go inside the CNS to the targeted site. So right now, the commercial products which are present in the market also are, most of them are the bioengineered one. And then we have the implantable matrices and like the hydrogels and all. So basically, we combine these two things, that is non-invasive intranasal route with the bioengineered materials together. So instead of sending these bioengineered materials through the oral route, sending them through the intranasal route is the best option, or the combination of the strategy, which many people around the globe are being actively doing. So approaches for the brain drug delivery, basically, if I talk about the intranasal delivery, it is a very convenient form because it is non-invasive method and it is more patient compliant. Patient can directly do it by itself. It doesn't have to have a very expertise thing that one requires. Then you have the direct movement of drug from submucosa space of nose to the brain into the cerebrospinal route, which goes on. And since you have the more moisture laden kind of nasal environment, so the the drug or the formulation that you prepare, it basically get absorbed at a very on a very fast scale with a good absorbability. And also this entire thing bypasses the first pass metabolism. So all in all, it's a win-win kind of route which one can opt for. And if I talk about the intranasal delivery, these are the same things which I discussed. Let us understand a pathway for reaching brain after intranasal delivery, how it reaches up. So once the drug or the biomolecule encapsulated in the nano formulation is being, sniffed through the um, through the nose, basically goes this drug goes and gets stick to the roof of the olfactory mucosa. Now it has the ciliated cells or the microvilli which take up Though it's a slow process, but still it takes up the biomolecule or the drug that you had transferred into the nose. Um, these globulate cells, they take it up and they just put it across the cribriform plate. It gets deposited on the cribriform plate, and from here it goes directly into the olfactory tract through the olfactory nerve, which pulls it up. Through when it comes into the tract or the olfactory bulb, we call it as commonly. So from here it directly goes to the brain, and that too into the third and fourth ventricle of the brain. So the areas there are your hypothalamus thalamic area. So these are the areas which for neurodegenerative diseases, they are the very targeted kind of areas. I mean, the entire pathology is basically happening in this particular region only. So it is basically giving you a lot of benefits. And if I talk about the assessing these strategies, it's been validated that intranasal drug delivery system is a better approach in many ways, the ways that we had discussed. Moreover, it also facilitates the direct movement of drug from submucosa space to nose to brain, and then getting easily dissolved in the CSF along with the fast drug absorption from the nasal cavity. So, all in all, it has the large vascular supply and quick onset of action by drugs. It also doesn't give you any side effects, and there are a number of advantages, particularly attractive, such as like being the highest increasing drug bioavailability and lesser side effects kind of advantage. So now we understand the mechanism. After understanding the mechanism of drug uptake into the brain from the nasal cavity, uh, basically we can understand. You must have understood that how it is going and it is being beneficial. Now, talking about the nano formulation, why now route is fine that it is for the direct targeting, but what is the use of nano formulation? Why it is needed? So to cross through the nasal route to reach cerebral cortex, you need to opt for the novel drug delivery carrier systems. So these are the strategies to deliver therapeutics to CNS, like there are like polymeric nanoparticles or solid lipid nanoparticles, maybe the implants in the cortical regions, nanogels, liposomes, and they do have a certain therapeutic limitations. Like in case of polymeric nanoparticles, they show very less thermodynamic stability and polymer degradation. Whereas the implants are like you had to go for the surgical interventions and all. Then you have the solid lipid nanoparticles. They also have their stability and release concerns. Nanogels and the macromolar structures, they tend to aggregate and they become unstable due to that. Same goes with the liposome. So all in all, it has been seen that the most appropriate approach or the most appropriate nano carrier system that you can combine with the intranasal drug delivery route is the nano or micro emulsion system. These are considered to be the better approach for nasal application as they are thermodynamically very stable than the other nano formulation, as its structural efficacy or efficiency enables the high drug loading capacity, high absorption along with good penetration ability of lipophilic drugs, and improved drug solubilization of hydrophobic drugs. So hence improving their bioavailability with its cost effective approach. So, and also it is very spontaneously formed with ease, and manufacturing and scale up at the industrial level is very easier in comparative to the other formulations. It also shows sustained or controlled drug release with excellent thermodynamic and kinetic stability also. So in this way, among all these strategies, um, till date, whatever the research has come up, the nano or the micro emulsion approaches are seen as a better approaches. Then there are other things also, the other approaches also, like the targeting ligands or modifying them, or the surface chemistry that can be modified. So even you go for the people go for, after making these nano carrier system also, they go for the surface modification by the conjugation thing, conjugating the surface charge, so that when it is electrostatically charged, it will go more towards the targeted side. So there are many various ways through which people are targeting the CNS disorders, and they are trying to figure out any therapy that can enable them to solve the root cause of the CNS disorder. One such study we are also conducting there in our lab, where we are going for the preclinical study in Alzheimer disease of the mouse model, where we are going with the intranasal delivery of curcuminoid, another nanoemulsion that we had prepared with the curcumin for amyloid and cognitive modulation, to split or to defragment the amyloid proteins and reverse the phenomena of cognition. Now, this we are doing with our industrial partner, Smarty Minds, in Western Australia. We are doing it in Flinders, and it is being funded by Dementia Australia Consortium. So, what we had done is we had prepared the oral application of curcumin, and it is the microemulsion system, sorry, nanoemulsion system that we had prepared. And the active compounds, um, we had taken the quantification of the active compounds present in the formulation, and trying it to pass through the intranasal route of the mice. So basically, we are doing the intranasal administration of this curcuminoid in the awakened mice, not in the sedative one, because we need to see the cognitive reassurance again in the mice. And after the treatment, it was being seen that, as you can see here, the results, it was being seen that around 40% of the cognitive ability was being restored after the treatment of curcumin nanoemulsion through intranasal route for a 4 weeks time period. We'll be again extending this study, and the entire, and how the intranasal is being given. Since mice is very small, so instead of the pipetting thing, we just did it with the catheter. The neonatal catheters, which are there in the neonatal ICUs, we basically insert it inside the olfactory region of the mice, and very slowly, it takes somewhere around 5 to 7 minutes to slowly pump in the formulation in both the nostrils, and then put the mice in an inverted condition so that it gets absorbed easily for another 10 minutes. And after that, we release the mice. So it was been seen that this particular method has revived back 40% of the cognitive decline which was there in the, and these were the genetically modulated mice, the ABP3 mice model, which where the cognitive decline was immense. I mean, so if we are getting that in the chronic stage of the dementia, the 40% reversal, uh, then it is then we are hopeful that around somewhere when you diagnose the Alzheimer at a very initial stage, that time if you go if you deliver this at that stage, then the percentage might go higher. Then the another important strategy, or the another study that we are doing, is with the theranostic efficacy of miRNA based thing, uh, based exosomal nanocarrier system. Now, exosomes are the biomolecules which has been released by all these cells in our body. So when these cells basically they release the nanocarrier, these extracellular vesicles, they are of various types. like they have the apoptotic bodies also, microvesicles also, and exosome is one type of the extracellular vesicles, EVs. So these exosomes, they act as a cargo package system. If they are being extracted from the cell lines or the cellular systems, and then they are being extracted, purified, identified, characterized, and then you put in your biomolecule, what the formulation that you had prepared, or maybe the any kind of biomolecule like your miRNA, into these cargo packages, the exosomes. Inside them you insert them over, and you reintroduce into the system again. So they being the biologically active molecule in the nanometric size range, they basically cross the BBB and they reach to the target site. So this is much more natural way of delivering the therapeutic compounds in the brain. So this we are trying up, and this is a more targeted approach that we had started up with. And then the other another study that we performed in the JIT Noida, along with the collaboration in BHU, was making the nano emulsion system for ginkgo biloba, the standard extract of ginkgo biloba, that is EGB 761. We made the nano emulsion of the same, and we tried doing the same, uh, the infusing the this particular formulation through the intranasal route into the mice. But here we want to see the overall accuracy, overall reversal, if it happens in any way in the mice. So here we had not taken the genetically modified mice, rather we it was a chemical induced scopolamine induced mice model, Alzheimer model. And we tried for the various tests like your Morris Water maze test and the Y maze test and the Western blot immunohistochemistry analysis, and acetylcholinesterase inhibition activity. This all was being performed, and it was being seen that instead of giving the oral, we also gave the oral treatment to the mice with the same formulation. And it was being seen that the intranasal formulation, the bioavailability of the intranasal formulation was 93% inside the brain tissues, in comparison to the ones which are there in the with the oral route. So this was the one difference that was being noted down. The other was that through the intranasal route, the reversal of the cognition part, and the disintegration of the amyloid protein was much higher than in comparison to the oral route. So this was the clear demarcation and a clear lead for us that yes, the intranasal route is working up, and we had to go ahead with this only rather than the oral formulation. And the other thing was that the reversal of the cognitive decline, that was something very much, that is what is being needed with these neurological disorder, if we can reverse on the phenomena. But we also saw that the neuronal damage that happened in a severe cases due to these plaques or the amyloid plaques there in the brain were not reversed back, showing that the cellular damage cannot be revived again, but it can improve on the condition, and at least it will stop the damage, further damage into the pathological site. So this was the pilot study that we worked upon, and the next work that we are doing is more of the detailed genetical and molecular analysis on the same thing, that what exactly is the pathway and the genes which are being involved for the reversal thing that is happening in the brain. So this is one set of thing that we had taken, and there are few. These are few publications that we just got through with a different type of nano systems, which actually can help us to get through the CNS drug delivery. And nothing can be achieved without a beautiful team that I have. And so I just want to thank and extend my gratitude to the team I work with here in India and in Australia, and due to which we are able to generate this much data, this much research data, and we can further go ahead with the research outcomes. And maybe someday, if we can go at the end and we can get some good leads in the research. So the only thing that I can convey at the end of the presentation is that translational approaches in the CNS disorder is now very much needed thing, and the gone are the days of the basic science work which was needed in the CNS, because all is being mostly has been explored and seen. Now is the time to get the therapeutic interventions done for the CNS disorders. So this is all from my side. If anyone has any query, any question, want to share any feedback. Dr. Gaurav, I had finished the presentation. Anyone has any query?
Yeah, yes, you also. So, dear participant, if you have any query, you can unmute yourself and ask the query. You can also write in the chat box, although you can also uh, okay. Kailash Chandra sir has asked, can it be prevented by the change of the lifestyle in modern era with proper clinical observation? Sir, Kailash Chandra sir,
Okay.
can you?
So he has asked for lifestyle modification. Yes, it can be, but see, once you talk about the CNS disorders, then usually lifestyle disorder, lifestyle changes and modification won't help much once you start seeing the symptoms of the CNS disorders, because it is that stage, if I talk pathologically, that in the end only, like crossing more than half the way, you actually get the signs and symptoms. If I talk about the Alzheimer's specifically, the signs and symptoms of Alzheimer are seen in the later stages. The pathology is going in your head, but they are being seen in the later stages. So yes, it may add on with the treatment, but treatment is the first and foremost thing that has been actually needed. It is not only with the like in the cardiovascular disorders, you can go for the lifestyle modification and you can control the disease, you can contain the disease. But with the CNS disorders, this is not exactly the same phenomena that will happen. You cannot contain or control the disease with the modification in your lifestyle, or maybe going in a more natural way, or the things. So that is one perspective.
Thank you so much, ma'am, for enlightening and this wonderful, informative, and insightful talk on translational
Thank you so
research
much,
in
Dr. Gaurav.
CNS drug delivery.
Thank you so much.
We are truly grateful to you for sharing your time, expertise, and invaluable insight with us today in this
It's fully a pleasure.
FDP.
It's fully a pleasure. Thank you so
And
much for
thank
inviting
you.
me.
So I would like to invite our Dean, Rana Patnaik ma'am, to kindly felicitate our speaker, uh, Manisha ma'am, with e-memento. Yes.
Thank you
Thank
very
you.
much, Dr. Manisha Singh, for such an informative lecture. And on behalf of SBMSTU and Innovation Council, please accept this memento.
Thank you. Thank you so much, ma'am.
Okay, thank you.
Okay. May I leave now? Thank you. Thanks a lot. Thank you, everyone. Have a good day.
Sudeep sir, I am not able to make you the presenter. Can you rejoin, sir? Am I audible to you? Okay, sir, now you can. Okay. Sudeep sir is joining again.
Dr. Godav, it is coming?
Yes, sir. Yes, sir. Yes, sir. Yes, sir. Good afternoon, sir.
Good afternoon. How are you?
Fine, sir.
Madam is also there. Pranam, madam.
Yeah.
Sudeep, I'm very excited to see you on screen.
Ma'am, first of all, take my regards and greetings from my side. It's quite fortunate that Gaurav organized this kind of event, and I'm quite nervous that I'm presenting in front of my supervisor and as well as in my lab junior. Both of you are there in the virtual mode in the screen. So, I have nothing to say about it. I'm quite nervous, ma'am. Ma'am, how are you, ma'am?
Fine.
Okay, ma'am. Okay.
Always busy here
Yeah.
and waiting for you to come and join this conference and all this.
Yeah. Obviously,
Participate.
Dr. Gaurav told me several times to come to Golgatias. So it is very hard time to go,
Hmm.
ma'am, and very difficult from Shillong to from anywhere in India to move. It's very difficult otherwise because traveling time is too
Hmm.
much. And okay, ma'am,
Hmm.
we'll go in near future. Uh, we'll start our session, Dr. Gaurav.
Ah, yeah.
Yes, sir. So it is
Start.
my honor and privilege to introduce our esteemed guest speaker, Dr. Sudeep Paul, sir. He is currently working as associate professor in the Department of Biomedical Engineering, Northeastern Hill University, Nehu, Shillong, India. And he completed his postdoctoral research at the School of Computer Science and Software Engineering, the University of West Australia, Perth, under the scheme of biotechnology, overseas associateship of the scientist working in the northeastern state of India, supported by the Department of Biotechnology. He received his PhD degree from the IIT BHU Varanasi with an specialization in electrophysiology and brain signal analysis. He also organized many workshops and conferences. Most significant are the Indian Academy of Neuroscience annual meeting, IEEE conference on computational performance evaluation in many years in 2020 and 2021. He is also organizer of IBRO APRC Associate School in 2017, IBRO Global Engagement 2021, and IBRO Meeting 2021, and 29th Annual Meeting of the Society of Neurochemistry India in 2015. Dr. Paul published nearly 60 international and national journal papers and more than 40 conference papers on different international and national platforms. He filed 8 patents and out of which 4 granted, and he completed 20 book projects in Springer Nature, Elsevier, and many more reputed publishers. Dr. Paul is a senior member of IEEE and also member of different society like IEEE, IS, AS, APSN, ISN, IBRO, and he received many awards, especially the World Federation of Neurology Traveling Fellowship, Young Investigator Award, IBRO Travelship Award, and ISN Travel Award. Dr. Sudeep sir is also contributed his knowledge in different international journals as editorial board member and reviewer. He has presented his research accomplishment in many countries like USA, Greece, France, South Africa, and Australia. So we are fortunate to invite you, sir, at this international faculty development program. Thank you so much, sir, for accepting our invitation, and we are eagerly waiting for your presentation also. You are muted, sir. Sir, you are muted. Sir, you are muted.
Thank you, Dr. Gaurav, and thank you, Madam, for giving us this chance to present this particular topic that I have chosen for this particular presentation. I saw that this program is basically the Faculty Development Program, FDP, and it is quite natural that when we are discussing about some research and innovation, these two terms is quite familiar at this moment, and all of you are very serious to do certain kind of research, and then we have to do how we can go further with that research in the innovation point of view, maybe in the biomedical science, biomedical engineering, or any other disciplines. But today we will discuss about the biomedical disciplines, biosciences, neurosciences. And so this topic that I have chosen, it is maybe different a little bit of your regular topic that is going to be delivered last 2 days and maybe in the next day tomorrow. This is IPR. Dr. Gora, my screen is visible?
Yes, sir, it is visible.
Okay, just confirm that the slide is changing. Just I am going for the slideshow mode. Just confirm me whether the slide is changing or not.
Sure, sir. Sure, sir.
The slide is changing, Gaurav?
Yes, yes, sir. Yes.
Okay, thank you so much. So the topic that I have chosen is today is the IPR and its importance in the research and development. This program, when we are talking about this IPR, so it's related with three very important terminology, that is the intellectual property right. So when we are discussing about the intellectual property right, that straight go to your research, innovation, and further the startup, entrepreneurship, and many more. So when we are discuss this topic in next 1 hour duration, we will learn a lot about this topic, because many researchers are doing research in their lab, but that particular product, that particular equipment, that particular material, that particular phenomena, that particular algorithm is there still in the lab, but it is not coming to the market. Unless and until it is coming to the market, there is a no fruitful outcome of your research. A research will be only fruitful when your product, your developed design will come into the market. Then you will be happy, your supervisor will be happy, your mentor will be happy, and ultimately our society will be benefited. As an Indian, we will be very proud for that particular fellow when certain product is coming to the market, like as a product and a prototype format. So, okay, without wasting of time, we will move further. So we will start with the intellectual property. So what is that intellectual property? I know that many of the faculties, students, researchers, those who are presenting in this or participating in this particular program, they knows about the intellectual property. But this, when this intellectual, one, what is the actual significance behind the term of intellectual? That means some kind of research that has to be done by yourself or by your team or by your colleague or as a group of team member, and when it is coming to a new form that is not there in the today's market or today's global scenario, that is the form of an intellectual. It is a property because when you develop certain nanomedicine, when you develop certain drug, when you develop certain kind of algorithm, when you develop certain kind of medical devices, Small device, no problem at all, but it is the property. Which property is this? Basic question is that. It is your property, your supervisor property, your group property, or your institution property, or it is the property of an India. So that is a very basic phenomena or question coming to your mind. And then rights, it's a right, because when you develop something in the lab, that should not be kept in the lab. It should come out to the road. So that's why today's a big highlight or big line has to be there. That is the lab to road. That means when you are developing certain thing in the lab, that has to be come out to the road. That means societal benefit, something come out. So that intellectual property has to be divided into three basic categories, that is copyright, patent, and trademark. So when we are discussing about the intellectual property, then something should come out in your mind that somebody are very, very famous for the music. Somebody are very, very famous for their some lyrics development. Somebody are phrases, words, comics, symbols, designs, inventions. So it cannot be a boundary. Anything can be done in this universe, and that can also be an property right. So that means when you develop certain phenomena in your lab or certain material in your lab, that has to be come out, and that has to be come out as a team of your lab, including of your supervisor and the total group member, like as a product format or as a simple, you can say the idea format, or you can say the phenomena format or symbol format. So that has to be coming out to the market, and you will be more happier than that day. So that thing has to be there. So we will discuss that what things you have go for the copyright, what things under patent, what things under trademark, because many researchers are doing research in the lab. But at this today's generation, they has to know that which things has to be done under which category, how he has to be done, what is the validity of their product. Everything or every product having some validity. When you purchase a medicine from the market, it has some expiry date. You cannot use the medicine after that thing. So when you develop certain kind of nano medicine or drug in your lab, it obviously having some short of the expiry in the form of a scientific expiry. Suppose I did something in my lab. After that, Gaurav was there. He did something in that particular lab. After that, XYZ person did something in the lab. So it is a generation wise the lab was developed. And here madam is there. So she was our mentor. So that means under the umbrella of Professor Patnaik, these things was carried out in a step by step. So that is the main phenomena of the. So that means when a researcher did something in the research, it should not be stopped at that particular moment. It has to be carried forward by the other researcher, then another researcher, then another researcher. So it is a chain system. But credit should go to the first researcher. That has to be done, including the supervisor and other team members of that particular time. So that means a primary right has to be classified into different category. So that is the copyright, patent, trademark, trade dress, trade secret, and geographical indication. So one by one we will go ahead. So first we are taking the copyright. What do you understand by copyright? Hello? So
Yes, sir. I am muting. I am muting.
yeah. So when we are doing certain thing of the copyright, it is an very important keeping in mind. Suppose you wrote your thesis. You wrote your thesis and that should have been copyrighted. Under which category? It is a copyright of your institution. It is the copyright of your university. It is the copyright of your particular things. Although the work has been carried out by you, by your supervisor, by your team member, but it's a copyright of the university. A copyright certificate has to be produced at the time of the submission of the thesis. That means it's a very important thing. that you copyright something. Now, when this copyright has to be theft, suppose you did something and another lab mate or lab junior or something like that, he copied the entire thing of whatever you made on your thesis. That is not acceptable at all. It should be an original work. That means thesis must be an original work. That's why plagiarism taking into the consideration under Shuddh Ganga. So everything has to be determined, or everything has to be checked whether this is a legal right of that particular person or not. If it is yes, then it is a very, very good. If it is not, you have to change. So this is the main phenomena of the copyright. And there are certain laws are there for the copyright. What are the major primary goal of your copyright? It has to be protected. Suppose you want to buy a software, Word, Microsoft Word. What is the license of that word? Microsoft. So Microsoft having its copyright. Nobody can purchase this software in the world apart from Microsoft. Otherwise, it is infringement or Copyright Act violation. And there is a huge loss or penalty that has to be. So that's why every system, whatever the software that you are using in the lab, somebody are using Origin, somebody are using MATLAB, somebody are using SPSS, somebody are using any format of the laptop software, that has to be original in nature. So that is the main important thing of the copyright. So that means thesis is one part, research is one part, whatever the software you are using, that is also a very, very critical or important role. Because in the thesis, in the methodology part, you have to be declare what version you have used in that particular work of that particular system analysis. So that has to be keeping in mind, and it has to be accepted by the reviewer. Sometimes reviewer asked whether what type of software you have used, that is licensed version or it is a pirated version. If it is pirated, then somebody can ask or somebody can question that from where you got this software. If it is an institutional license, then no problem at all. If it is a personal license, no problem at all. If it is the license will be taken care from your other sources, XYZ, without the knowledge of that particular company or the person, then that is the violation of the copyright law. That has not to be accepted. So as a researcher, you have to be very, very careful what software you are using, that is absolutely free or not. If it is not free, then this software is pirated or original. If it is original, keep the license with you. That means the product key. That has to be very, very crucial at the end of your submission and award of your PhD degree. And sometimes when you guide your students as a teacher, as a supervisor, the supervisor can buy the software and every student can use that software in that particular lab. So it can be a lab wise also. So that has to be manageable by that particular thing. That is the main important terminology of the transfer of copyright. Suppose you buy something and you have to transfer that copyright to others. How you do that copyright transfer? There is a special format is there if you go to the Indian Patent Office website, then you will find it out how to transfer the copyright from one person to another person. Suppose you made certain algorithm and if X person made something and Y person want to buy it, so they have to transfer the copyright under some certain rules and regulations. So that person can buy the software and can buy the algorithm, can buy these things. So that is under the copyright transfer. Then copyright law in 1957, first Copyright Act was there under the copyright law in India. In 1958, 21st of January. Today is 21st of February. So today, the 21st of January 1958, this Copyright Act was first implemented or applicable. That means it was there in the first in 1957, but applicability has been done after a year in 1958. And since then, this kind of things has been amended. Every time it is amended, those who are teachers, they know what is the amended. Every time UGC amending their rule in 2015, 16, 19, 18, now 22 rules has come. So every time it is amending, copyright law also it is amending day by day. For what purpose? The purpose is to protect your invention. Purpose is to protect your innovation. Purpose is to protect your research in a very, very systematic manner. So that is the main important motto of the government, to take the initiative and to save your product, save your things, save your research in a good fashion. Now we can go for the duration. What is the duration of that particular thing? And what are the basic things that under that copyright? Copyright, suppose you design something, that is under copyright. That is the literacy, dramatic, music, artistic work. Somebody very, very interested to draw something. Painting, that you can do for copyright. Suppose you made a lyrics of any song of your own. You make a poem. You wrote a poem. You wrote a story, that has to be done under the Copyright Act. So you can do the copyright. So that is the main important thing that I want to highlight in the today's domain in the field of the Innovation Council. So basically, it is the second, the sound record, government works, public undertakings. So everything has to be done. So different ages are there. That means that validity. I told you the validity. When you purchase a car, what is the validity of the tax duration? It is 5 years, then 15 years. That is called lifelong. So that's why everything has a validity. So it is the validity of the author. Author means, suppose you are the researcher, you as the author, plus 60 years from the beginning of your research or year, the next of these things. So this is the validity of these particular things. So now, when we are considering this, the copyright order in 1991, it is a very important thing that the term of copyright in a work shall not exceed that which you enjoyed by its own country of origin. Very, very important line. Suppose the Indian researcher from Golgotia University invented something that cannot be done from Australia, New Zealand, any part of the other global country, that cannot be the same thing will be done as a copyrighted. So it is a violation. You have to do your own country first, and then you have to file to the other foreign country with the same product. This is a law, and you have to follow that law. So when you do something, this kind of research, you have to file the copyright in your home country first, and then you have to file to the other country. But you cannot straightforward go to the other country and file the copyright. It is a violation. Because the work has been carried out in India, not in US, not in Australia, not in England, not in Japan, Singapore, nothing. So you have to do the first in India, your home country, then you go for the other country. Somebody having a collaboration work. So what you do that kind of collaboration? You have to do your primary supervisor or primary researcher, leading author from which country. So you have to do this thing from that past, and then you go for the other country. So then there is some fair use. What do you understand by the fair use? Fair use means when you do something, that means the purpose and the character of the use is including whether such use of the commercial nature or non-profit educational purposes. I told you that when university or institute purchase a license or purchase certain phenomena algorithm for your purposes, suppose a researcher required MATLAB software. So from where they will purchase the MATLAB? They will straight intend from the either from the university or from their pocket from the MathWorks. So that means they are the non-profit educational purposes, that software has to be there. It is the nature of the copyright work is very, very systematic in nature. Very, very systematic. That means when you declare your invention, it has to be clearly written. Because if you change one sentence, if your own development or own work, the total meaning of your invention will be changed. So suppose you invented something but copyrighted something else. So it is some kind of dilemma can be occurred in the near future. So you have to be very careful. Whatever you made it, whatever drug or whatever material you are making in your lab, so that particular material you have to be go for that particular inventory purposes. Now public domain, what are the things can be there? So when you design something and it has to be public domain. Suppose you wrote the thesis, that will be in the public domain under Shudh Ganga or any other portal, no problem at all. But unless and until your thesis is totally not published, suppose some figure, some equation, some tables, some graphs, some plots that is still remain published, not published. So you can stop there. You can request them not to upload that particular chapter, or you can lock the chapter in that particular duration, and that has to be done. That is there in the copyright law. So unless and until the author of the particular work is fully satisfied, the material don't use to the others, it is a violation of the copyright. Now coming to the patent. So it is all about the copyright. Now coming to the patent, what you understand by the patent? Patent is basically a device. Patent is basically your material. Patent is basically your product. So this is the patent. Copyright is basically different thing. I told you, it is a official work related. Your thesis is copyright, but your thesis is not patented. So patent will be done your actual work, but thesis will be done under copyright. So the patent actually is for the 20 years validity. The patent validity is 20 years from the date of the application. Please keep in mind, not the date of the grant of your patent. Somebody can confuse the 20 years from the date of your grant certificate. No, it is the date from where you have applied first day. That is the date, that is called the application date. From that date, the patent will be valid for 20 years. That means 20 years it is your product. And after that, it can go, you can commercialize it within 20 years, you can sell it, or anybody can, or you can go for the startup, you can start your own business. Nowadays, many institutions, many researchers are doing their own business and developing their product. So you can go for that thing. You can go for the, you can sell your product like as a small device or material or any kind of things. So that is under the patent. So now there are patent office, if you can see, there are 4 different metro cities are there. 4 different metro cities having the patent office. Main patent office in Calcutta, in Delhi, in Chennai, and in Mumbai. So there you have to be there. Under which jurisdiction your patent? Suppose you are from Golgotia, so you have to apply your patent in Delhi. Suppose I am in Shillong, so I have to apply in Kolkata. So there is a certain jurisdiction is there. So under that jurisdiction, the patent office is there. You have to apply under the patent office of that particular product. And there certain step-by-step guidelines is there. So they are Controller General of Patents, then Design and Trademarks, then the different sections are there under that patent office. So they will do that kind of thing. And after that, it has to be amended. You can see that every 2 or 3 or 4 years, it has to be amended. 2003, 5, 6, 12, 13, 14, 16. So every quite frequently these rules and regulations are amending. So you have to be keep on watching the what is the recent rule of doing certain kind of things under patent. So you have to be checked when you do something under patent, because you are doing research in your lab, but that product is not coming out. So for these things, you have to be continue watch or continue search on the patent office website. You have to download the form, you have to download the regulation rules, or you can take help from your patent cell of your particular institution. If you go for a sector-wise analysis, if you can see, you can easily find out that the biotech patent sector is less comparison to the all other sectors. Just see, that is the biotech is only 5%. The patent was done Indian. It is Indian. Please keep in mind Indian. Then chemical is 18%. Drug is food is 13%. Electrical related is the 6%. Mechanical is 18%. Computer related is 14%. Other means it can be of the anything, device development or something like that. It is a 26%. But straight biotechnology related is only 5%. But drug is there is the 13%. So that means it is a quite natural that we are in little bit slow down of this particular application of the patent, and we are not taking serious effort for doing certain kind of things that what is developing in our lab that has to be coming out to the market. This is the snapshot that I took for your easy reference. That is the main snapshot of the IPR website. When you just Google the IPO, that is the intellectual property right website, IPO, IPR. So then this paper or page will open. In that particular thing, you will see the patent, you will see the design, you will see the trademark, you will see the GI, that is the geographical index. So these particular things you will see. Under the tab of these things, you can drop down menu, you will find out everything, the forms related to what purpose, forms related to which purpose, everything you will get it from that under the each tab. So no need to worry about it. And then you can find it out the copyright offices there, and then the journal. And suppose you when you publish your research in a good journal, Elsevier, Springer, any journal, Nature. So what is there? After publication, that the journal will give you a copy. So same thing over here. When your invention will be published under the this patent office, so it will come under the Indian Patent Office journal. So your invention will come in the Patent Office journal web page. You will find it out, and you can find and you can cite your research from there. So that is called the journal. This is the journal. So you can find it out from here. That if you click, then every Friday, generally if it is not a holiday, then every Friday this journal is used to open. So that means every Friday new version, new edition, new issue will come out. So you can follow it up after application. Now coming to the trademark, I hope many of you are quite familiar with this terminology, trademark. Suppose you are buying one Coca-Cola bottle, you will find it out this TM, that is called trademark. Suppose you buy a new Hyundai car, Hyundai logo, and after that you will find this trademark. So basically trademark is used for this particular design, logo, sign, expression, this kind of thing you can do the trademark, not for the device. Trademark is basically used for your labeling, packaging, this kind of thing. This trademark is generally used. Somebody can give the name of the particular concern. Suppose ABC, a company name. So ABC, a company name or something like that. So that has to be marked as a trademark. And at the top of these things, it is written TM and a circle. So that means it is a trademark of that particular company. Or the company having some logo. So every company requires some sort of logo. Every institution is having some logo. That is under trademark. Nobody can use in this universe in the same name. So it will be violation of the Trademark Act. So that is very important. Now trade dress. What is the trade dress? Trade dress is basically a legal term Act generally refer to the characteristics of the visual appearance. Visual appearance, that is also an important thing. What type of appearance you are having, that is signify the particular things of this thing under this trade secret or trade dress. Now trade secret, I told you the Coca-Cola. So this is the Coca-Cola, and you know the Coca-Cola, I told you this is the trademark, that the name of the particular company is trademark, but the how the bottle will be and how the logo will be pasted in the bottle, that is called the trade dress. That is also comes under a certain invention. Every institution is having an innovation council. They are doing a very hard job, or they are doing day and night job for supporting the students to design something new, and they are also supporting how to go further for patent, copyright, trade secret, trademark of that particular things. You can do that thing. Suppose you made some material and you want to give some name. The design of your material is under the patent. Name of your material is under the copyright. And the when you make a total things, that is under trade dress. So different categories are there, and you have to follow that thing, particular things. So everything when coming into in different locations of India, all of you know that Darjeeling is famous for obviously for Kanchenjunga, but apart from this Kanchenjunga, there are very famous is the Darjeeling tea. Assam, very near to my official place, Shillong, that is Assam is also famous for the Assam tea. Obviously, there are so many places are there to visit in Assam, but the Assam tea is the blend mode, that is called the geographical indication. Geographical indication means in the geography, in the map wise, in particular location, in your location, which type of things is important, and that has to be a indication of your particular things. So that has to be very important. So if you can go for the picture, you can see that which type of material is under GI tag. This is called GI tag. When you do certain kind of things or purchase, so that particular things is there for that particular area. So every Indian places, if you can see the red dots, so you will see the name of the product from that particular places are famous. It is called the geographical indication tag. So that means this sector is famous for this particular type of material development. It can be a MSME or it can be a startup, it can be entrepreneurship, that is a different concept. But output is the product that I told you in the first slide. Don't do your research totally in the lab. Come out to the market, come out to the road and serve our nation in a good form. So that's why it is come as a geographical index. Now, few slides left, near about 10 slides left, that I will talk about the importance in research and development. How this IPR is important in your research and development. First is the fostering innovation. Everybody know that we are developing something and it has to be coming out to the market. So that is the encourage. Encouragement has to be there to each and every research scholar, that the research and development should be encouraging. And then facilitating collaboration and technology transfer. Suppose Dr. Gaurav is doing very good in Golgotia and I am doing here, certain kind of mutual collaboration we can do, and we can facilitate some kind of thing. We can make certain things over here and he can also do certain thing over there, and we can mutually transfer to each and everything technology. So both of you are beneficial for technology transfer. This is called technology transfer. So for these things, both of the party are enjoy, both the party will enjoy the flavor from that development. Second is the insensitivizing investment, that is the attracting funding. Suppose nowadays MSME or your innovation council will give a very big poster that come and join to the startup, or come and open to your entrepreneurship, promote entrepreneurship. First prize is 50 lakh rupees. Come out with your idea and you present your idea and you can earn the 50 lakh rupees from your idea. That has to be there. That is called promotion. And then obviously the fair, ensuring fair recognition and reward. That means the judgment, what is best and what is the better and what is good. That means first, second, third. Good is third, better is second, best is first. That means you have to be best category. Then a fair judgment must be there. And your product will be marketable very soon. That is called commercialization of your product. Then balancing innovation and public interest. So when you are doing certain IPR, that is intellectual property right, then it should be increase the state economy, national economy, because nowadays our government is giving the money to the institution, but they are also telling at the same time, start build up some kind of teamwork for self-sustainability. The self-sustainability is very important for research and development, especially those who are researcher. Because whatever you are developing from your lab, you have to earn or you have to commercialize the product and earn some money to survive that lab and to survive the lab equipment and all. That means the government will give money to set up the lab one time, but after that, from the revenue generated after commercialization, you have to survive. That is called sustainability. That self-sustainability in each lab is very important. Then dozens of industries across India, our economy, rely on the adequate enforcement of the patent, trademark and copyright. From where? From ours, yours, as a teachers, as a student, researcher. Your development, your product, your research will come out as a product, trademark, copyright. Then the company will earn some money because you are selling something to the company and company will sell double of the buying price. So then company will be beneficial, and that IP also safe. There is a safer side of the IP because no need to bother about the IP at that particular time. That is called IP, means intellectual property. Third is the without protection of ideas, business and individuals would not reap the full benefits of their inventions and would focus less on the research and development. Very important. Similarly, artists, those who are their artists, they can also do certain kind of work apart from your research to concentrate on the inventory part of things. That is also a fruitful result of your near future. Fostering innovation, I already told you how to do fostering, how to do manage your research, how to do awareness of your research. So that has to be there. It should be a chain system, and every chain has to give some sort of output, like as a patent or copyright. So now see how many copyright or patent will come out at the end of the year or at the end of the day. So to give you the best result as an Indian citizen. So the fostering innovation, like as a technology transfer, is also very important in form of a collaboration between the academia and industry. Suppose you have a facility, full facility of your research, but you don't have a facility for a big industry to work. So if the industry will help you, if industry will wants to help, then you can make a mutual collaboration, like as a MOU, that is Memorandum of Understanding. You can make and you can do certain kind of good thing, like as a technology transfer between academia and industry. Then only you can do certain kind of commercialization, further development, business and workflow. You know that when we are talking about certain disease, and then from where the disease recovery, suppose the disease recovery medicine will come out. It is coming from actually from the lab, academia, but we are purchasing the medicine from the industry, like different pharmaceutical industry, because it was a technology was transferred from academia to industry. So this is very important. So when we are doing certain, everybody having idea, when you implement that idea and when you sell that idea to in the form of a technology transfer, so that has to be there to make a good shape of your innovation. Then incentivizing the investment, funding. Nowadays everybody is running to the funding. Funding scarcity is there. How to maintain it, how to maintain our lab without funding. So it is called as a self-sustainability. So when you make certain product, suppose you will go after 5 years or 6 years from your lab. So, but what type of product you develop now? So your supervisor will be thankful from your research to take certain funding help after commercializing that product from the for developing further development of the lab. That is self-sustainability. Financial resources are very important, but it is a safeguard of our investment, because you don't know the future. But if you have the money, then nobody can stop you. Nobody can stop your research. Nobody can stop your invention. Today, the discussion about the breezing of the research and development in the biomedical sciences. This is the breeze. This is the actual breeze to develop. This is the actual breeze to transfer. Because that one side is researcher, the opposite end of the breeze is the investor or industry. So there is a breeze. So when you cross the breeze by using the technology transfer, then it is a fulfillment of your research. So for these things, what kind of things is required? Entrepreneurship. That will be taken care by your innovation council team. You can take help from your innovation council's team of your institute and do something best for these things. That is called promoting entrepreneurship. Fair compensation for creator, I already told. Very important is that the balance innovation and public interest. When you did something in the lab, it is very important note that the intellectual property is not absolute. Those mechanisms such as patent expiration, which allows invention to enter public domain after a set of period. Please keep in mind, I told you, after 20 years, all the IP will be public interest. Same thing, a project is for 3 years, but after the completion of the project, all the asset will be under the particular institution's property. Although you can use it, but it is the property of the institution. But money has come from the government. Suppose DBT, DST has given the money to purchase the equipment to run the project. The same thing over here, when you are doing something, try to be the technology transfer, commercialization, or the further step of the upgradation of your patent, copyright within the prescribed period, that is the 20 years, sometimes 5 years, sometimes 10 years. So that has to be taken care. So fair use provisions of the copyright law, which enable limited use of the copyrighted material for the purpose like education and research, achieve a balance between encouraging innovation and serving the public interest. So that means the educational institutions are more aware at this particular moment that they want their more product, they want the more innovation, they want the more copyright, and that's why the Innovation Institution Innovation Council, that is IIC, is continuously working on that, and they are doing this kind of continuous awareness program. For what? For maintaining a particular balance for the betterment of the institution as well as the society. So thank you so much. I hope all of you got some idea about the IPR. It's a little bit different topic from that particular FDP, although if you don't know about the IPR related things, I hope it is not a at the end of the day you have a something in your mind that we develop a lot of things in our lab, but after that what happened? After that nothing. But don't feel like that, where you have a good thing in your mind, so you can be a billionaire in the near future. Thank you so much. Thank you.
Thank you so much, sir, for your expertise in this field of intellectual property rights, provided us with a valuable insights into the significance of protecting innovation, creativity in the area of research and development. This in this era, it is also called. It will be not exaggerate if we called the war. This is the era of innovation.
Yeah.
The ministry is also focusing on the to promote the entrepreneurship and innovation at India.
Yeah.
And your understanding in the IPR is the important for elevating the intellectual property rights at any campus. And we are truly grateful to have you as
Sure.
a keynote speaker. Thank
Sure.
you so much for sharing your time, expertise, and wonderful session. The valuable insight into the patent, copyright, trademark, GI tag, and all the thing related to IPR. So once again, thank you, sir, for outstanding presentation and for being an encouragement for us in the field of IPR. So,
yeah, I know
dear
that you are
participants,
also a good active member in IIC at Golgotia.
yes, sir.
So
So, at our university, we have established the IIC, sir, in the academic year 1819. And
yeah.
from that, we have received the highest rating, that is 4-star rating from the Ministry of Education. And this
So
year, we are
a big
also
clap for the
included.
Gaurav. Yeah.
Yes, sir.
Yeah.
We got included in the IIC consortium of Uttar Pradesh as a governing body member. And in this year, the ministry also selected us for the funding, with the funding support, to mentor the 5 institutions. And we have selected 5 institutions from the Uttar Pradesh, including Gautam Buddha University, SSV PG College Hapur, Tikaram Mahavidyalaya Aligarh, Shobhit University, and RGPG College Meerut. And we have
If
also
I'm not
initiated the
wrong,
mentorship support.
Dr. Gaurav, if I'm not
Yes,
wrong, in the last year, when you organize certain program, I told you to deliver certain lecture here at Nehu. That time you were so busy for that IIC at Golgotia.
yes, sir, yes, sir.
So he was the one of the leading member there. So it is very nice, very nice. And I am very happy that my junior is doing this kind of great activity, and very nice. I'm feeling very happy.
Thank you
And
so much,
we are
sir,
as
for the blessing and your kind word in
every
your.
blessing is coming from madam. So she is there, my supervisor, my mentor from masters. And if she is not there, then we are totally helpless. So
Yes,
every
sir.
blessing is coming from their her end till today. Yeah.
Actually, our academic has been born after the supervision of the ma'am. So,
Yeah, yeah.
yes, sir. So, dear participant, if you have any query, you can unmute yourself or you can put the message and question in the chat box. We have 5 more minutes for the question answer. I'm also allowed you. You can unmute yourself also. If you have any query related to patent, copyright, trademark, GI, design patent, sir has a great expertise in this area. So please take this as an opportunity. If you have any query, you can unmute, please.
ये उन्हीं की है, उन्हीं की फोटो आएगा, GUSC ACC की हमारी फोटो की, हमें अभी फोटो करो तो आई है।
If you don't have any questions, you can track my mail ID. I have already given, or you can take it from Dr. Gaurav from the team, and you can mail me. So I'll be very happy to answer. If you have any question, any doubt in your mind related to IPR, and so I'll be very happy to share with you. And so my basic request is that now the era is the product has to be come out to the market, so not being concentrated to the lab. So lab to road connection must be there. That's why this program is there, bridging the development, research and development in biomedical science. Yeah.
So in the chat box is flooded with the thank you and very excellent session. Sir, the participant is only writing in the chat box, sir, and providing the feedback. So there, I am not able to find any question. So how to reach out the speaker letter? So Anuj, I will forward the email ID of sir later. If you have any query, you can ask related to IPR. Okay. So I would like to invite our Dean, Professor Rena Patnaik ma'am, to kindly felicitate our keynote speaker with the e-memento. So this is the digital presentation. So sir, kindly receive the e-memento.
Thank you very much, Sudeep, for such a nice, such an informative lecture. And I am really mesmerized. I'm so happy to see you here. है ना? I'm happy to see my students growing like this. है ना? बहुत जम के आप लोग आगे बढ़ रहे हो. I'm बहुत खुशी है मेरे को अपना फैमिली जो है इस तरह से बढ़ रहा है, ऊपर चढ़ रहे हो तुम लोग, हैं सुदीप? And on behalf of School of Biomedical Sciences and Galgutia's University, accept this momentum.
ओके, थैंक यू सो मच मैम. मैम आपका आशीर्वाद में मैम और आशीर्वाद दीजिए जो आगे जाए हम लोग सब, तो अच्छा लगेगा. मतलब फाइनली it is your proud moment that as a child we are growing further for the research and development, and we are very sincere that Gaurav is there, myself and other many students are there in foreign, so they are excellent work. You are a you are a like as a diamond in BHU. When I was there in from 2007 to 2012, that is a proud, means the golden time I ever in my life. So thank you so much, ma'am. Thank you.
Thank you, Sudeep. Actually, I think God has blessed me with such nice, wonderful students. My family.
Yeah.
You all are the members of my family.
Yeah.
All of you are so good. That is why our lab flourished.
Yeah. Yeah.
So
Yeah.
I always thank to God for
Yeah.
this.
Yeah.
All the complete chain koi kam nahi hai, all are at the top.
Yeah.
I'm happy.
Yeah.
Happy
Thank
to see
you.
you all
Thank you.
here.
Thank you.
And my blessing is always there with you all.
Yeah. Thank you.
ठीक है?
Pranam, madam. Pranam.
हाँ हाँ ठीक, नमस्ते.
Thank you, sir. Thank you so much, sir.
Thank you. Thank you.
So our next speaker has also joined. Uh, Good afternoon, Dr. Yuvnes. And it is my honor and privilege to introduce our invited speaker, Dr. Yuvnes. He is an assistant professor in the Department of Applied Mechanics at MNNIT, Allahabad. Dr. Yuvnes received a BE degree in Biomedical Engineering from Anna University, Tamil Nadu, and MTech from the NIT Rourkela, and awarded with the PhD degree from IIT BHU Varanasi. After his doctoral degree, he worked as a postdoctoral fellow in the Department of Engineering Design at the Indian Institute of Technology, Madras, Chennai. Dr. Yunus got prestigious awards like DST Serb National Postdoctoral Fellowship in 2021, Sitar Bayrak Gandhian Young Technological Innovation GYTI Award in 2019, Frugal Innovation Award from the Bayrak Government of India under the Sitair scheme, and DBT International Travel Grant in 2018, and many more. So far in his career, he published uh, more than 40 articles that can include 5 books, chapter, 27 journal publication, 8 conference article, that is indexed in the SCI database, and he also hold 2 Indian patent. So I am very fortunate to invite you, Yuvnes, at this international faculty development program. Over to you.
Thank you, sir. Thanks for the brief. It's quite big. And actually, I don't hold 2 Indian patents. It's yet to receive the patent grant. So just a correction. We have received EFER, first examiner report, and it is we are about to submit the response to it. So remaining things, it is okay. Being a senior, it was quite natural
So,
to be honest.
now you have corrected.
Yeah.
So it is all right.
Okay, sir.
Now,
Shall I share?
yes, you can share.
Is it okay, sir?
Yes, now visible.
Good evening, everyone. Myself, Dr. Yumnesh, currently working in a system professor at MIT Allahabad, is going to give an talk on novel cell-based biosensing device for cellular analysis. So thanks for the invitation, sir. So
Thank you.
these are the course contents, which will be including. I will start with the basic to the deeper end. So from introduction, origin, various types, then comes our cell-based biosensor. What are the various structures we have designed and we have developed? Then I will be concluding with concluding the session. So going further, so introduction. Can anyone define biosensor from the audience?
I think
Okay,
audience cannot
fine.
do not want to unmute themselves. They are very active
Okay,
in
okay.
chat box.
Okay, I can't able to see. Okay, as for IUPAC, that is Indian Union of Pure and Applied Chemistry, biosensor is defined as an analytical device that uses specific biochemical reaction mediated by isolated enzymes or immunosystem, tissue, organelles, or whole cell to detect chemical component, usually by electrical, thermal, optical property. This is a classic example, standard example for biosensor. So this is a schematic representation of the biosensor. where usually it is designed by integrating a biological element with a physiochemical component. Physiochemical component to provide a quantitative analytical information of a biological response in terms of an electrical signal. The physiochemical component here I have written as physical and chemical. We can combine and we can tell physiochemical component can be a transducer or a detector element to detect and analyze by converting the biological response into an appropriate signal for a specific interaction. After a specific interaction, when it combines, it converts into an a specific signal. Okay, the signal generated is converted into measurable electrical parameters such as current or voltage through a suitable transducer, which is processed and displayed in a readout. So the biosensor generally consists of three parts. First one, it is like a component with recognizing the light. Second, produces a signal, and third, analyze and produces a signal. Second, it is signal transaction. Third is a reader device. Fine. So origin in 1962, C. Clock, Professor C. Clock carried out his first experiment research to detect oxygen. We are using that what you call the pH meter, right? So same kind of setup he fabricated, and he tried to detect oxygen using a platinum electrode, and he has successfully done that one. So it is marked as the origin of biosensor. The year the contest, what generally happened is that it has what he did is he immobilized the platinum electrode with glucose acetate. It is an enzyme. So when it interacted with the glucose, it converts, like I have shown, right? When it get interact, it will produce gluconic acid. Then oxidation and reduction cycle will happen. Then we will be getting H2O2 and O2. From there, it further the byproduct get divided, and we can able to sense the excess of hydrogen atom present in that. So more hydrogen atom, so more sugar content, more oxygen is assumed by measuring H plus. Okay, higher the sugar content, more oxygen is assumed, and the lower the content, more the hydrogen peroxide is produced. This means either an increase in hydrogen peroxide or decrease in oxygen can be measured to indicate the glucose concentration. Fine. This is the schematic representation of what Professor Clark has designed with all the chemical equation I have shown. So next, based on the transaction principle, the biosensor has been classified in broadly in three types. First, electrochemical, then optical, then acoustic wave. Acoustic wave or piezoelectric biosensor, we can tell. So going into deeper, electrochemical biosensor, it is nothing. Transducer, it is nothing but electrode. Here, the electrode itself will act as a transducer element in the presence of an analyte. So the electrochemical biosensor detect the change as a change in voltage or current in real time by coupling an electrically sensitive layer, that is an recognition element. The electrochemical biosensor build up of either 2 electrode or 3 electrode. We can able to see here. Either it will be like working and reference, or it will have counter electrode, reference electrode, and working electrode. Like this, we will be having a system, and we through and potential stat, we can able to see the changes, the change in energy, basically. So next, amperometric sensor. So amperometric biosensor measures the current produced by redox reaction that occurs at an electrode surface when a constant potential is applied. The current is proportional to the concentration of analyte. Here it is more toward the current, which we need to emphasize the amperometric, as the name says, right? Ampero. So here it is like more towards current. So basically, we have glucose meter and all, right? In the home, it is basically an amperometric biosensor where we will be having enzyme in it. It will be like 3 electrode system where where they have immobilized glucose oxidase upon and over so when the glucose from the body, so the blood interacts with the glucose oxidase present, it produces gluconic acid and byproducts. So based on the H plus concentration of oxygen, our blood glucose will be the range will be given. Okay, it is just an example. So amperometric biosensors are commonly used to detect gases such as oxygen and glucose. Gases, fine. Then the other class is that potentiometric biosensor. The potentiometric biosensors, the measurement is based on potential difference. Here, potential means voltage. Here we will be measuring the voltage difference between the two electrodes. So one electrode is usually reference, and other electrode it is working. So where the reaction with the analyte occurs, so the potential difference is related to the analyte concentration. So these biosensors are often used for pH and pH measurement or ion detection. So we can able to see here we are using 3 electrode system for finding the current. Here it is more than enough to have 2 electrode working and reference to measure the voltage difference. That's it. So generally, for measuring the intra and the cellular response, right, where we can insert an micro pipette electrode into the cell, and with respect to outer, we can able to measure the action potential. It is a common classic example.
सीधा है, जो बच्चा खुद से लिखेगा या कहीं से देख के लिखेगा तो आई कांटेक्ट से पता चल जाता है।
ऐसे भी बदतमीजी कर रहा था, ऐसा नहीं करना होता है।
Can I move further?
Muted now.
Okay, fine. So conductometric conductance, it is coming, right? The every keywords it is present over there at the title. So here the conductometric biosensor measure the change in conductivity. Conductivity is nothing but we know that current, if we our houses it is been having power supply, right? When we touch, due to condensation of electricity into our body, where we will be acting as an ground, we get shock. So same thing, the change in conductivity of the solution due to presence of analyte. By measuring that change in conductivity, we will be so we can tell how much concentration of that analyte. For example, in pH, if H plus is high, we will be having more conductance. Whereas in OH, that the basic region, there the basic OH will be more, so we won't be having that much conductance. It was one of the example. So basically, conductometric biosensor measure the change in conductivity of the solution due to presence of analyte. The conductivity is typically measured between 2 electrodes in the solution. The change in conductivity is proportional to the concentration of analyte. These biosensors are used to detect various ions, molecule that affect the conductivity of the solution. So then impedometric biosensor, impedance, right? The opposite. Impedance means it will oppose the flow of current, oppose the flow of current. So impedometric measures the impedance, that is that opposes the flow of current of an electrolyte interface. The impedance is influenced by presence of analyte, which causes change in electrical property of the interface. These biosensors are often used to detect molecules like DNA, protein, and cell. For example, here we have cell, right? We can assume, we can give an electrical equivalent of this biological cell, where the cell membrane, right? it is selective. It is a bilipid layer. It is selective for some ion which is positively charged, and it will oppose for some selective layer, right? It acts like a selective layer. So the cell membrane, right, we can as it is selective, it can store and discharge, so we can relate that with an capacitor. Another, the intracellular, right, it is nothing but ionic concentration, right? So we can relate it with an impedance, sorry, a resistance. And extracellular, we can again represent it resistant, and it can be represented by these setup, where we have cell membrane capacitance, intracellular fluid resistance, and extracellular fluid resistance. Simple. So these when we seed a cell on this substrate, based on their coverage, we can able to see change in the current. By measuring the change in current, we can able to plot the dielectric change in dielectric behavior of the system. So this is one. So then optical. So again, I hope students were not able to see. Right. Molecules are analyte. These biosensors are based on the principle of optics, and typically involves the interaction of light with an sensing element and the analyte of interest. For example, you have a substrate on which, for example, we can have a colorimetric sensor. Okay. For example, colorimetric means the color will change based on the interaction. If we take a glucose, we can tag that H plus. For example, if we have phenol red is highly sensitive to the change in pH. So change in pH means H plus concentration. So more the oxygen, the color will change. So it is one way where we can use optical. So we can change, we can sense by using passing a light, we can able to see at which nanometer the light get deflected. Okay, this is one example. So we can what we can do, we can immobilize an antigen. When antigen antibody interaction takes place, the change in concentration happen, which will affect the light intensity. So by measuring the change in light intensity, we can able to correlate with the concentration of analyte. So good example is that we have a UV spectrophotometer, right? So we can by using the light, we can able to see the spectrum, right? That is one example. So surface plasma resonance. Surface plasma resonance generally uses a gold film. So based on the interaction of for example, antigen antibody, the surface of the gold will get changed. So by passing a light against it, we can able to detect the change in spectrum. So change in spectrum in terms of response. So surface plasma resonance biosensor uses a thin metal film, especially commonly gold and silver is used on a glass substrate. When the light is incident on a metal film at a specific angle, it excites plasmonic surface plasma oscillation of free electrons at the metal surface. The resonance angle is sensitive to the change in refractive index near the surface, such as those caused by binding of sunlight to the sensor surface. By measuring the change in resonance angle, the presence and concentration of sunlight can be detected. For example, here I have shown you the C image, right, where we can able to see when it is not been interacted, when there is no the antigen, so antibody, there is no reaction. When association happen, when it get interact, a change in a response happen with respect to time. When as the concentration based on the concentration gradient, the interaction will end to certain point, right? After that, dissociation take place. Then we can it will get regenerated. Means by flowing and neutral pH or something, we can remove all the changes and we can bring it to and back. So here we can measure the change in spectrum. So the surface plasma, it will absorb the surface plasma will generate only at the particular node. For example, if we have gold nano particle of 560 nanometer, and if you excite the gold nanoparticle with only 560 nanometer, it will get it will absorb that radiation. It will totally absorb all the light source, and the electrons will move to the from valency band to conduction band. Then as it attain maximum, heat will be generated, and heat will be generated, and due to and it will come back to the valency, and due to returning back of as the heat get eliminated, it will come to the valency band. So fiber optic biosensor, here it is like we will be having the tip will be coated with some antigen and antigen. So when it reacts with that particular antibody, the change in the spectrum will be causing. So generally, fiber optic biosensor uses optic fiber to transmit and detect light. The fiber are often functionalized with a sensing material that interacts with the analyte. So the change in refractive index or the fluorescence property of the sensing material due to analyte binding can be measured by detecting the analyte concentration. So here we have light source. It will go through the optic fiber, and based on the interaction, the change in spectrum, the change in the intensity will be detected. The refractor signal will be detected, and we will be based on the changes what happen the resonance, the refractive index or the property of the material will change. By measuring that, we will be determining the analyte concentration. It's a simple. So fluorescence same. The fiber optic will have a fluorescence particle which will be particularly sensitive for and target. So when antigen antibody reaction happen, the fluorescence signal will be emitted in presence of a light, the targeted light. For example, in MTD, we used to use a I'm not right, 460 nanometer. So during that, the MTD is sensitive to that.
I think due to the poor network, speaker, I'm contacting them.
हाँ कर लिया मुझे कब ज्वाइन करा कब हटा था
Welcome back.
So I will reshare.
Yes.
What I will do, I will share the window. So from where it got disconnected?
Approximately 5 minutes before. Okay.
Here?
Yes, yes.
Okay, fine. So what I will do, I will start with, again, I will start from here. So electrochemical transducer, the electrode itself here, the electrode itself is will act as an transacting element in the presence of an analyte. So here we will be having an working electrode. For example, people uses carbon rod and working electrode as a working electrode, and counter electrode we need to use a platinum, and reference electrode it is AgHCl. So for example, if we are immersing it in a pH, the to sense and uh, since the different pH solution where the concentration of the pH will get changed, will be varied, the working electrode itself will act as an transacting element. It will sense the change in H plus atom, so H plus ion. So which we can through which we can able to determine, or we can tell that this much concentration of analyte is present in that solution. So biochemical, so electrochemical biosensor detect the signal as a change in voltage or current in in real time by coupling an chemically sensitive element. For example, we can use glucose oxidase or cholesterol to detect cholesterol. The electrochemical biosensor build up of either two electrode or three electrode system. So amperometric biosensor measures the current produced by redox reaction that occurs at an electrode surface when a constant potential is applied. The current, ampere means current, so the current is proportional to the concentration of the analyte. So the amperometric biosensors are commonly used to detect gas such as oxygen and glucose. So potential means voltage. So here we are going to measure voltage difference. So potentiometric biosensor, the measurement is based on the potential difference between two electrode. One electrode is usually working electrode, and the other is reference electrode, where the reaction between the analyte occurs. The potential difference is related to the analyte concentration. These biosensors are often used for pH measurement or ion detection. So conductometric biosensor conductance. Conductance means electrical conductivity. So conductometric biosensor measures the change in conductivity of the solution due to the presence of analyte. The conductivity is typically measured between two electrodes in solution. The change in the conductance is proportional to the concentration of analyte. For example, if you are using pH, same I will relate everything with the pH. So for example, if you have pH in when the pH is 1 or something, the concentration of H plus is very high. So over there the conductivity will be high. Whereas in basic OH will be there due to which opposes the flow. So these by which we can able to differentiate when we use different pH, we can able to see what pH gives us what type of conductance, what type of value. So the change in conductivity is proportional to the concentration, of course. So these biosensors are used to detect various ions or molecules that affects the conductivity of the solution. So impedance is nothing but flow opposes the flow of current. So impedometric biosensor measures the impedance of an electrode electrolyte interface. The impedance is influenced by the presence of analyte, which causes the change in electrical property of the interface. These biosensors are often used to detect biomolecules like DNA, protein, and cells. Because what we generally do is that in impedometric biosensor, we won't be supplying the sensor more than 100 millivolt, and based on the safety regulations of human body, and human body, it is okay to supply us with 100 100 millivolt, which is nominal, and it won't cause any effect, any burning effect or tingling effect into our cells, ourselves. So it is being supplied with constant current. So change in current we will be measuring, by which we can provide we can come up with an impedance versus frequency plot. So generally, for example, and we can relate the output with an equivalent electrical circuit. For example, here I have given an typical example of an cell, biological cell, where we can consider the cell membrane. It is selective permeable membrane, right? It can be related to selective permeable because it allows some molecules freely in and out, and it blocks certain selective ions. So based on this property, it is generally considered that the cell membrane it is related with an capacitor, where generally in electronics we can able to see charging and discharging effect. So then the fluids which are present in and out, it can be related with the resistor. So it is the equivalent circuit of this one. So when we have the impedance data, when we fit this with the existing with an electrical module, we can able to see the change in capacitance, change in resistance value. Theoretical, for theoretical examination, we can use this. And optical, so the classic example of optical biosensor is we know that gold nanoparticle we can use, right? So when the gold nanoparticle is being immobilized with a certain antigen, when it get interacted with that particular antibody, the it will cause the antigen antibody will cause in the presence of light, it will affect uh, the refractive index. Okay, so refractive index Ri. So refractive index based biosensor, these biosensors detect change in refractive index of a sample when an analyte binds to the sensing element. The binding event causes a change in local refractive index, which can be measured using the techniques such as surface plasma resonance or informatic. The refractive index based biosensors are highly sensitive and can detect analyte in real time without needing the label tagging. We don't need tags. Okay, so optical biosensor offers several advantages, which include higher sensitivity, real-time monitoring capability, and the ability to perform measurements in complex sample, because here it will be based on antibody and antigen. So they are widely used in various applications like medical diagnosis and environmental monitoring and safety. So optical biosensors are device that uses light to detect the presence or concentration of biological market or analyte. These biosensors are based on the principle of optics, and typically involves interaction of light with an sensing element and the analyte of interest. So here we will be supplying with a certain nanometer, and after the antibody antigen reaction, we will be getting an change in wavelength or refractive index. So this is the mechanism, and when we use gold nanoparticle, it will be having an absorption spectra of particular wavelength. So that time at that wavelength it will absorb more amount of light, that is more energy. We can use so hence it is being used like that. So for another one example is that where you would have been seen UV spectroscopy, right? So over there we can use a different concentration of gold, and we can able to see how the intensity get changed, or silver of particular nanometer when we use antigen antibody reaction or some byproduct. For example, we can for example some drug, the release study, drug release study, the drug will have an absorption at particular wavelength, so we can able to see from a scaffold at that particular time. For example, when we see when we drop and after some hours when we take the byproduct and we see how the spectra, we will be seeing the spectra, the release kinetics, right? So like that we can use this one. So really then after 4, 7, 14, 24 hours we'll be seeing the release of the drug, and we will be drawing the graph to see how the release has been, sustained release has been happened, right? It is a classic example of an optic biosensor, especially for UVs. So it is nothing but we will be seeing the absorption spectrum at what particular wavelength the absorption is high. So as I told about the refractive index, the classic example of refractive index is surface plasma resonance biosensor, where we will be having uh, on the glass we will be having a gold coating or silver coating. So the gold nano, the gold film will be sensitive for a particular wavelength. So when it got excited with that particular wavelength, the reflectance light, right, the wavelength of the reflectance light will get changed. For example, you can able to see from A it got changed to P, so based on the interaction. So and when there is a flu, when even though when there is no and particularly in this principle it is like, even though we have analyte in the presence in that medium, if we are not passing the light it won't react. So what it will do like is that when we apply the light, the gold nanoparticle will absorb the light and it will conduct from the valency band. The electron will move from valency band to conductor gamma at max when it attains after getting reached to valency band, because it took a certain ignition, right? So after that what it will do, it will release the stored energy in the form of heat, or energy, and it will comes to valence band again. So this change, right, we can able to do it. So in the presence of so simple, when it get associated with an antibody and antigen, we will see a change in a sharp change in the response. And when we stop the light, it will get dissociated and will come to the natural backend. Backend. So the what the it is a working principle. Now we'll be seeing what does it typically say. So surface plasma resonance biosensor uses a thin metal film, commonly gold or silver, coated on a glass substrate. When the light is incident on a metal film at an specific angle, which is used to excite that particular film wave thing. So surface plasma oscillation of free electron at the metal surface occurs. The resonance angle is sensitive to the angle, to the change in refractive index near the surface, such as those caused by binding of analytes to surface sensor surface. By measuring the change in resonance angle, the presence and the concentration of analyte can be detected. So fiber optic here, what people generally do is that they will be coating the tip, right, which is being interacted with the analyte. Over there, we will be coating the tip with an particular antibody antigen. So when it get interactive with the antibody, due to the antigen antibody reaction, in presence of light, the spectrum, right, the refractive index get changed. So due to by measuring the change, we can able to plot the graph which can be correlated with the concentration. So fiber optic biosensor uses optical fiber to transmit and detect light. The fiber optic can often functionalize with a sensing material that interacts with the analyte. Changes in the refractive index or fluorescence property of the sensing element due to analyte binding can be measured to determine the analyte concentration. So similarly, as like the fiber optics, here the with the fiber optic we will be coating the fiber optics with an fluorescence material. So when the target, when it get interact with the target molecule, the fluorescence molecule now, its intensity or wavelength it get changed due to the interaction between antigen and antibody reaction. The fluorescence get released generally. So these, for example, if you have gold is tagged with gold nanoparticle is tagged with fluorescent material, and on the top when it is been conjugated with a particular what you call antibody. So when antigen antibody occurs, it get released the outer surface form of plasma, the energy increases due to the heating, the dissociation happen, due to which the molecules which are present, the fluorescent molecule which are present near in the gold will get excited. So typically what here we will be doing, we will be checking it by passing and particular wavelength. So these biosensor uses a fluorescent molecule as a sensing element. When the target analyte binds to the fluorescent molecule, it causes change in fluorescence intensity or wavelength, which can be detected and correlated to the analyte concentration. So luminance based biosensor, we can another term is that photoluminance based biosensor. We can also do. For example, here what we can do is that we can take our body as an example. So we can ask a patient to have a cup of gold particle tagged with an particular antigen, and when it get interacted to, for example, for seeing the cancer, for visualizing the cancer marker, we can tag that one with the cancer marker, and we can give it we can feed them. So by passing light to a particular, we can able to attract, or by placing an magnet over there, by placing a magnetic field around the body, we can able to see, we can able to analyze the quenching effect or enhancing effect generally. So so the biosensor uses the luminant material that emits light when excited by an external light source. The presence of the analyte can be quench or enhance the fluorescence signal, which can be measured to determine the analyte concentration. So infrometric biosensor, here we will be having different core, means optical fiber with different core. So here it can be like we can have an reference and then target one also. So both as the concentration changes, the each and every individual core will give us with some a reading change in wavelength in a real time, in one set time. So in a particular time, we can able to compare the with the reference one. This is the basic behind that one. So informatic biosensor uses in inference pattern generated by the light wave to detect change in the refractive index near the sensor surface. Analyte binding causes change in the interference pattern, which can be measured and correlated to the analyte concentration. So colorimetric biosensor, for example, if we have the phenol red, phenol red is very sensitive to H plus ion, H plus and OH. When the concentration of H is very high, it will give us the reddish in color. When it is very less pH, means more pH, that is 11 or 10, more than 7 or 10, the color get changed to yellowish. So by seeing the visual color change, we can able to see, we can for pH paper, right, we can able to tell that at what pH it is being there. So colorimetric biosensor are type of biosensor that detect the presence or concentration of the target analyte by producing the measurable color change. These biosensors are based on the principle that the interaction between analyte and the specific recognition element lead to change in optical property of the chromatogenic indicator or reagent or indicator, resulting in visible color change that can be detected and quantified. So next is scatter based biosensor. The scattering based biosensor rely on the interaction between light and the particle in the sample. When the light is incident on the sample, it can be scattered in different direction depending on the size, shape, and refractive index of the index in the sample. By measuring the scattered light information about the sample, including the presence of analyte, can be obtained. So scattering based biosensor are used in label-free detection of biomolecules and nanoparticle based biosensor. So here the detection principle is based on interference between okay, interaction between the analyte and the particles in the sample. For example, if we use Raman, for example, if we have a graphene based item, so graphene where G band, we have two G band, D band. D band intensity will get changed based on the interaction. So this is an example where the scattering can be used. So acoustic, it is emitting nothing but sound waves. Acoustic biosensor are type of biosensor that use a acoustic wave to detect the analytes as a biological molecular cell. The working principle of acoustic biosensor involves the interaction of acoustic wave with target analyte, which causes measurable change in acoustic signal. So, for example, we have piezoelectric substrate. So, piezoelectric substrate, and when we pass or when we excite that due to dipole effect, the acoustic, the mechanical signal, right? The acoustic signal, it will be generated. The electrical energy, it will be converted into mechanical, then the acoustic signal, which we can tell it as acoustic. So when we coat the substrate top with an biomolecule, as the concentration or as it get dissociated, the change in concentration happen. So based on that, our what you call our concentration, we can correlate that change in mass with the concentration of the biomolecule, through which we can say that if it is for we need to create a lookup table, which is we call as a calibration plot, through which we can able to map that one. If this much concentration of biomolecule is there, it get released, then we will be having this much reflector point. The spectrum get changed, right? So surface acoustic wave biosensor SAW, where it is as like that, we will be having here, we can able to see on the substrate, we will be having an idea interdigital electrode, where we can, it is like a springy kind of, which is physically isolated. When we excite that with an supply, the electrical energy, it will be converted into mechanical energy due to dipole effect, and based on the concentration, we'll be getting a change in the acoustic current at the receiving end. So surface acoustic wave biosensor uses piezoelectric substrate to generate and detect acoustic wave. When an electrical signal is applied to the substrate, it generates acoustic wave that travels along the surface. These waves are sensitive to change in mass or stiffness on the surface. When the target analyte bind to the surface, sensor surface, it causes a change in the acoustic wave velocity, the flow, right? Velocity or attenuation, which can be measured or correlated by the analyte concentration. So bulk acoustic wave biosensor. Here we will be using quasi-crystal microbalance. It is nothing but and crystal when, for example, when the surface of the crystal is being occupied by the mass, change in mass, we will be getting an output as a change in frequency. This is a classic instrument which we use it in for used in an closed container for measuring the amount of coating. For example, we need to coat, for example, I have a watch band, I need to coat it with pure gold. So what I will be using, I will be using a thermal deposition machine where I will be placing my watch stab over there, and I will be applying current at the bottom in order to evaporate the gold. So here we will be having quasi crystal micropropeller. So the amount of mass it is being changed, we will be getting in terms of change in frequency. Here we can able to see as a mass get increased, we see that change in the frequency, right? This is the basic working principle. So acoustic bulk acoustic wave biosensor uses a thick piezoelectric material as a sensing element similar to SAW biosensor. An electrical signal is applied to the material and generate acoustic wave. The wave travels through the bulk of the material and changes the change in the acoustic property, velocity or attenuation due to binding of sunlight or measured. So summary, so far we have seen various type of biosensor available sensing variety of molecule. I have shown a few, for example, antigen, antibody, and pH changes, but we can go on see a variety of things. So the question to the audience is that is it possible to use these biosensor for cellular analysis? Yes, we can, but with some modification. So now we are getting into the presentation, the actual theme. Before moving into that, why the cell-based biosensor is needed. So commonly used a technique to monitor cellular proliferation and viabilities uses a fluorescence spectroscopy, flow cytometer, and single point assays such as WST1, XTT, MTT assay, and BRDU assays. These are all a single point assay where we will be killing the cell functioning, and the byproduct, right? We will be using that byproduct to correlate with the cellular concentration. These traditional method used this traditionally used method of monitoring cellular transformation mentioned above furnishes us with either quantitative or semi-quantitative data apart from its requirement of labor-intensive protocol and expensive equipment. Moreover, these cell-based assays greatly compromise the consistency and reproducibility of the experiment. In addition to the distribution and in distributing the normal visualized state of the cell, resulting in their destruction. Therefore, to precisely monitor the cellular behavior in an external environment, a real-time, non-invasive and label-free method is desirable. Typically, the biosensor can quickly detect various diseases, thus dramatically reduces the heavy load on regular clinical diagnosis in the healthcare industry. That's why it is widely explored in the field of pharmaceutical, environmental technology, and medical sciences. So now it is sure that when we use something different, we can some biosensor, we can eliminate the killing or destruction of the natural environment of the cell culture. So cell-based biosensor, what it is definition? Cell-based biosensor directly uses the live cell. So we will be having the sensor. We will be what we will be doing. We will be replacing the analyte or the sensing membrane, right? We'll be using replacing the sensing layer with housing the sensing layer with the cells, direct cells. So due to when we add any analyte or target of interest, it will affect the cell. So when it affect the cell, the layer which is there, right? The transducer which is there on which the cells is sitting, it will change its dielectric property. So by that way, when we measure the signal change in signal, we can able to come up with an idea that what affected the cell and what kind of changes it is doing. So by correlating, we can correlate that change in electrical signal with an optical signal, optical image. So cell-based biosensor uses live cell in the direct biochemical effect. The biochemical effect of the direct partial contact of live cell is converted into quantitative electric signal by sensing layer, that is the transducer, which is present beneath the cells. The signal comes to the transducer to the various phenomena due to various phenomena such as change in ionic transportation, metabolic pathway, membrane potential, barrier resistance, and morphology of the cell. So this is a schematic representation of the cell-based biosensor where we'll be having cells present directly housed on the transducer. So any physiochemical property which affect the cell, due to the stimuli, our transducer will produce an proportional signal in the form of change in voltage or current. So by measuring the change in voltage or current, we can able to come up with what stage the cell is there. So different types of cell-based biosensor which are available is, for example, here we have a two electrode system. So the principle is like the current finds the closest spot. So here we can able to, it is been housed in such a way that the current gets uniformly distributed. So when there is no cells available over there, it is like few free flow. So there won't be any change in current absorbed. But when there is the cell, we can able to see when it get adhere, during the adhesion, less number of current will be disturbed. When the cell as it proliferate, the more it proliferate, the more it will affect the flow of current. So the working principle is simple, is that the cells which are present on the electrode surface affect the flow of current. So by measuring the change in current flow, we can come up with the occupancy of the cells present. So these sensors we generally tell it as electric cell impedance sensing system. So now comes the cost distribution. There are different types of sensors available. Minimally, it will be costing us around 15 to 20,000 per experiment. We can able to three here. They had eight sets, so we can perform 8 different experiment at a time. Because once it is been taken out, we can't. Once it is being destroyed or something, again next time when you reuse it, it will be giving us contamination result and other things. So it is basically prescribed to use single time. So this for per experiment, if we are spending 15,000, means it is quite costly. Even though this instrument which is used for detecting the changes, right, it itself will be giving us 20 to 30 lakhs per, and it is reusable. But this one per experiment we need to spend 15,000, which is very costly, so we can't afford to it. So we come up with replacing these biosensor. So later we will be seeing that one. That type of cell-based biosensor is that, as like we have seen, right? Surface plasma resonance. What we can do is that we can insert, we can use the same device with the cell. So as it is been interacted with an analyte of or external stimuli, there will be a change in refractive index. By measuring that, we can able to come up with an idea that what made, based on the concentration of this external stimuli, we can come up with an idea that how as the concentration increases, how it is being affected. So same like that, for example, here It is the principle of cellular activation by ligand. It is same. So we will be activating it, and based on the change, based on the interaction, we will be having the shift in the spectrum. So by measuring the shift in spectrum, we can come up with an idea how much antigen, antibody, or whatever what type of drug has been, based on what kind of concentration, how much cell being affected. So electronically also we can compare, we can detect the changes both way. Third type it is simple. We will be having a piezoelectric material based on the change in concentration, the frequency, right? The oscillation will get a change. So by measuring this oscillation, we can tell that the how much mass has been deposited on the crystal. Simple. So for example, here we can tell based on the adhesion, as the adhesion is more, the flow will get reduced. So change in distance, the deflection, right? We can correlate it with the mass. So design and development of novel cell-based biosensing device for cellular analysis. Give me a minute. So inspiration, right? We need to have certain inspiration. We know that, right? While going through a road or something, we can able to see red color light. When the night it will start blinking, but in the daytime it won't blink because it is been attached with photodiode and solar system, where during the light time the light will the proton energy from the light from the sun, it will absorb and it will store it in the battery. In the night when there is no light flow, the battery charge to the battery tend to display the charge. So that time we will be having change in we will start to change in current in the form of the light blinking, right? It's one type of it is an example of flow example of absorbing the light. So same field principle, when the light is being removed on the detecting surface within cell, based on the occupancy, the increase based on the change in the surface, our current, the flow of current will get a change, right? So which we have used. So for example, what we have done is like by taking a photodiode as an example, it is a typical structure of metal, insulator metal, where we will be having an insulating material as a center. So this is an one type of structure. For example, we have resistor, right? We can assume the resistor will have an insulating material inside it, which opposes the flow of current. So what we have done, we have replaced that one, this structure like this, where we will be having metal at the center, we will be having insulator, and at the end we will be having so metal. So this will be then at the top of the insulator we will be replacing that with an electrolyte. So why metal oxide? When the surface of the metal oxide is exposed to any particular external environment, the variation in the electron charge transfer is detected either in the form of change in resistance, capacitance, or impedance due to change in dielectric property of that material. Dielectric property, bulk electron mobility, and gray boundaries, bulk oxidation material, etc. So what it gives us is that cost and time, and transparency and biocompatibility, and the metal oxide provides. So when the metal oxide is being exposed to certain environment, there will be a change in resistance or capacitance or dielectric as a combination. Dielectric means a combination of resistance and capacitance. So by measuring this dielectric property, we can come up with the variation, how much variation it has been affected. So this is a fabrication procedure which I used. For example, we will be having ITO. We can for creating the contact, what we can do, we can selectively etch out the unwanted region. After etching out the unwanted region using SCL, we can deposit the, for example, here I took aluminum oxide as an aluminum oxide as a sensing element, and we can paste and culture cell culture holding cap, and after that we can immobilize it with gelatin in order to improve the cell adhering capability. So this is a structure of photograph of the fabricated metal insulated metal biosensor. So after that, what I have done is that we have seeded 3,000 cells, and we have checked how, with respect to time, how it get, how the cellular morphology get changed. So we have used, as it is providing us a transparency, so it gave us a possibility to visualize through microscope. So here we can able to see around 4 hours it is more round in structure, whereas from 8 hours when it got enough ECM production, so it adhered and it started to take its natural structure. As the time goes on, it started to adhere properly, and here we can able to see cell to cell interaction started happening. This is because when the area get reduced, right, when the mass get increased. So this is the image from the sensor through optical microscope. Similarly, we have checked is impedance it electrically. So here we can able to see how the capacitive effect of the sensor. We will be using that sensor in an semiconductor parameter analyzer, and we can able to see the black line. It is fabricated, and in presence of cell, in presence of culture media, when it is 8 hours, we can able to see it is here. We can able to, there is no capacity here, but when it is around 72 hours, you can able to see there is lot, the capacitance of that instrument, it got increased a lot. So similarly, with the capacitive behavior, the impedance, which impedance, it is nothing but flow, the resist the flow of current, right? So when we see this one, we can able to see as the cell, it get the cell of mart get increases on the surface, we can able to see the sensor started to offer us maximum resistance value. So with respect, it is a reason why the 72 or spectrum is at the top of everything. When we compare phase, it is nothing but for example, the leaking effect. You have cell membrane, right? A cell membrane, it will act as a capacitive element. It can charge and discharge. So based on the morphology, right? The morphology, the change in morphology affect the phase of the system. So by this we can able to see the angle get drastically reduced. It shows that there is enough change in the morphology has been happened. So impedance, it is nothing but it is so, and the Nyquist plot will give us, it will the phase, it will give us the morphological change. Impedance, it will give us the occupancy. Capacitance, it is the behavioral, and impedance and the Nyquist plot, it will tell us the stability, how the structure is being modified. So summary, so now it is sure that we can able to compare the optical and the electrical research. We can able to see as the cell starts to proliferate, and when it starts to proliferate over the function of time, it affects the typical characteristics of the fabricated sensor. So these results demonstrate that dielectric behavior of the myoblast cells. I have used the C2C12 cell is capable of altering the typical and the predefined characteristic electrical behavior of fabricated in vitro cell substrate device. That is nothing but our metal insulator. That main insulator, it is nothing but here we have used Al2O3, so aluminum oxide. So further the MIM, we can use the MIM structure to determine various cell functions such as cell adhesion, spreading, proliferation, differentiation. The effect induced by the cellular function behavior is well correlated with both change in characteristic, electrical property, and microscopic observation of the cell. Hence, mRNA structure can be used for various cell-based biosensing applications. So now, as we what we thought, okay, we have stuck using insulating, which insulator means it will offer more resistance. What happens when we use semiconducting material in place of that? So similar structure we took. So we replace everything, and this is an this one is Zetano. Here we have used Zetano 0.2 mole. This is a detailed solvent preparation. After preparing it, I will be coating it using spin coater. So after coating the substrate, the conducting surface using conducting a glass substrate using the Zetano salt. First, we need to understand the characteristic behavior of the coated film, right? So what we have done, we have used XRD and we have matched with JSPS. JSPS, what the it showed a typical JSPA database, the whatever the film which has been fabricated. It showed a JSPS file of 00089139. It particularly matched with an approximation of zero point zero 0.01 nanometer difference, and degree difference. And after that, for cellular adhesion process, we need to know how rough the substrate is. So using AFM analysis, we measured the roughness. The average roughness of these 0.2 mole Zetano solution found to be 11.08 nanometer. After that, what we went for FESM in order to see whether it is continuity, it is continuous, the surface morphology generally. So through here we can able to see it is smooth and smooth without any discontinuity. So with the FESM we have done XPS to confirm the whatever is being fabricated is of zinc. It is of zinc oxide. So as we use silicon glass material, as we used glass on the top on which we have coated the Zetano thin film, the the silicon it is showing up. So in the maximum, if you see that we can able to see through which we can confirm that whatever it is been fabricated, the film, it is been we can able to confirm it is Zetano film. So after that, I went for UV spectro to see what is the range through which we can able to see the energy band, energy observe. So it is found to have energy of 3.3 electron volt, the film which we fabricated. Then what we did to as we got inspiration from photo detector, we wanted to see whether it is sensitive to the UV light or not. So what we kept under the dot, we have measured the value. Then we exposed that with UV light of 365 nanometer for 15 seconds. After that, we turned off the light under the light luminous. We have checked how the current characteristics get changed. So it is from this result we can able to see the when we are supplying the instrument, the sensor with -1 to 1 volt, it is giving us -2 microampere to +3 nearly +3 microampere under the influence of UV light. So this shows that the Zetano thin film which we are fabricating is sensitive towards light. So we have similarly as like MIM, we have fabricated the same precision, but here what I did, to in order to improve the cellular adhering capability, what I did, I have surface functionalized with gelatin. So it is like we have OH right on the top of Zetano. We can functionalize with amine group. So I use APDS. After that, I have done group cross linking in order to get aldehyde group. Once we have aldehyde group, we can incubate it with gelatin so that the gelatin will go and adhere on the will have an problem. So this after everything, it is like we need to See, right here we can able to see the gelatin, right? Due to surface modification, the amendments which are needed has been present. It shows to FTR analysis. Then after that, what we did to in order to know whether it is compatible or not, what we did MTT assay. So here we can able to on the glass substrate we have seeded the cell, and we have Zaterno thin film and surface functionalized. We can able to see how around when it get when the time increases, the cell proliferate more on the gelatin functionalized one than that of the other, and this shows that it is compatible. So then what we do, we seeded the sensor with 3000 cells 3000 cells per well, and these are the results. Here we can able to see till D it is a 72 hours, the we can able to see cells. After that, due to inherent behavior that the concentration of cell is very less, and the Zetano can offer us adverse effect here. It is it has antimicrobial and antimicrobial effect, right? Due to which it became toxic enough. So after certain time, it started to kill the cells which are present. So till 72 hours, we observed an increase in cell, and after that it got decreased. So we need to have a certain result which is repair, which is correlating with these. So electrical measurement, when we checked, we found that around 72 hours here, it is showing a good current response. After that, it got decreased because here in the impedance vectors we can we can see here we can able to see around 72 hours the impedance, the flow of resistance, the flow of current is being drastically reduced, due to which our impedance is very high. And after that it got 96 or it got reduced as the number of cells, the cellular mass is very high, very low. So similarly, due to change in morphology, we observed a change in the phase. And here through Nyquist plot, we can able to see how the dielectric, how the system been affected by the increase in the cellular mass. So here with the same time, I have shown how the cell number it is been changed at a particular wavelength, that is 10 kilohertz. So then what we did, actually we could not able to use it for long duration of time. So we increased the cellular.
Sorry to disturb you. Actually, the time is already past. So
Okay.
Please conclude.
Okay. Here we can able to see as the concentration increases around 24 hours, we can able to see good number of cells. So till we can able to see the cells are good enough till D, that is 96 hours. After that, it due to cell-to-cell interaction, and as the cell as the environment which is available for more to get proliferate, it is it got reduced. The cells started to detach. After it becomes uniform, that is like a tissue, it got tissue like formation monolayer, it got formed, then it got detached. So similarly we need to have a result which is emphasizing this. So here we can able to see still I have here 144 hours. So till that we can able to see there is a gradual linear increase in the impedance, which reflects which tells us as the cellular mass get increased, it get increased as the cell got detached from that substrate, our the impedance got reduced. So summary, the result demonstrate that dynamic behavior of the myobacter is capable of altering the physical predefined characteristic, electrical behavior of the fabricated in vitro cell subsistence system. Further, the emergence is also be used to determine various cellular functions. So, and the effect induced by the cell function behavior is well correlated with both characteristic electrical property and microscopic observed. So yes, we can use the MSM structured device for the various cell-based biosensing. Sorry, I took quite a long time.
Okay, I am highly thankful to you for accepting our invitation and delivering such an exceptional talk on novel cell-based biosensing device for cellular analysis. And you have did a lot of research in this area, but due to the time constraint, I am apology for that, that I have stopped you. So your expertise and insight benefited the participant a lot, I might think. And your discussion on this novel cell-based biosensing devices was not only enlightened us, but also are very inspiring. And you are a dedicated researcher, and you are working to advance the field of cellular analysis through the innovative biosensing device. It is and performing your work, a very worthy work. So, and your research and contribution also have the potential to revolutionize the way we approach for the cellular analysis, as well as this type of technology can be used and have a various application for the numerous field like biotechnology, medicine, and biomedical engineering. So I am highly grateful to have you as a speaker in this session. So now
Sir, I
this is
have
the
one question. One of Dr. Satyajit Tripathi has asked a question. Is it possible to check membrane surface charge of alive cell with the morphology change or during different adhering condition in vitro culture? The thing is that generally cells are negatively charged, as it offers an selective membrane kind of thing. So, and when we have the same negative potential at the bottom, the cell won't be adhering properly. The cells will be alive, but it won't take the proper shape, which, for example, fibroblast, it need to, we can able to see, right? It need to take some shape. So when it is like that, we can't able to see when it is opposite same charge, then the cell, it won't take what you call the proper shape. The cell will be like alive and it will be round. You can able to see increase in the cellular number mass, but the shape you can't see. Yes, what we can able to do, we can use an positively charged substrate or surface modification technique in order to facilitate it with proper shape. That's how we can able to measure through. After that, we need to use face or face. By measuring the face value, we can able to get the change in morphological information. I'm clear. When it is same, the morphology it won't take, so we won't be getting the morphology information. It will be same for whatever time you do, but we will be having change in impedance value due to increase in the cell mass. But when we have different, or when we do surface modification, the cell structure it will get changed. So we will be having change in morphological value in terms of change in phase of the impedance.
great
I
work,
hope it is fine.
great explanation, and very wonderful session, Eunice, indeed. And thank you so much for this insightful presentation. And I am requesting you to kindly accept the e-memento as a gratitude of honor from the Galgotia University.
Thank you, thank you, thank you, thank you, thanks a lot, sir.
No. So, Doctor,
Okay, thank you so much, Eunice. Thank you. And dear participant, I have
sir,
sent the feedback form link in the chat box, for which you
sir,
have messaged a lot. So uh, please
sir,
have a look. Yes, yes.
Someone is
sir. Actually, you have sent the feedback forms for day 2, sir, not 3.
The date, it is date 3.
No, yeah, actually the link is day 2 is available in the
No,
chat
it
box.
is date 3. You can fill this.
Okay, sir. Okay. Because it is mentioned as 2.
No, it is date 3.
Okay. Fine, sir. Thank you.
Gaurav sir, I cannot find a link of your feedback form. I'm still searching on the set, but I didn't find. Actually, I'm way to home and joining from the phone, and my battery is having only 5%. So please, if possible, try to send on the WhatsApp group for today itself. I'm requesting, sir, for please today itself send in the WhatsApp
Okay.
group.
Okay, you
Thank
can
you, sir.
message me, ma'am. I will share.
Okay, sir. Okay, sir.
Okay.
Thank you, sir.
Thank you.
Another 1 minute, sir.
And Meenakshi
For
sir,
unmute,
you are very welcome and for this FDP. And again, we have met from the
Yeah, yes, sir.
faculty
Yes, sir.
development
Nice,
program.
nice to
Yes,
meet
sir.
you, sir. Nice to meet
Thank
you. But
you.
actually,
Yeah.
I cannot because the topic is known to me. But only thing is the department is biomedics. No, but it is a I belong to a management domain. So
Okay.
I am relating the topic to that particular one. That is why I'm not able to converse, create the doubts on these things. Anyway,
So
very
actually,
nice to meet you, sir. Very
yes, sir.
nice to
Thank
meet
you,
you.
sir. So this
Welcome,
FDP
sir.
has been
you
organized
can
in the
relate
area
into
of
the town
biomedical
basic.
engineering. Yes, yes, please.
Yeah,
Meenakshi.
yes, sir. Yes, sir. Because
We can,
I was having an experience with pharma marketing. So to some extent, I can relate to these things.
Okay.
it was like we started with fabricating a low cost